Okay, good evening, everybody. I'm here. Hopefully, y'all can hear me okay. I just came from Art New Style stream. What's going on, BGS? You said on your left? Okay. I don't see you over here. I'll put the link in the chat as well. But tonight I was gonna speak with AM1. I sent him the link earlier. I don't know, I don't see anyone in here right now. Let me see if I'm on. Let's see if I'm on, and I'll continue. Yep. So I'm on. What's going on, DJ Moss? Y'all hit the like button on your way in. So I sent the link out to AM1. I'm just waiting to see if he's here. He might be a little late or something, but that's okay. I can just keep going. And the basic idea that we were going to discuss is Koch's postulates. I did a video, I put up a video the other day, and of course it was about COVID, and he had some comments in the section below, the comment section, and he mentioned, like, did they use Koch's postulates to, I guess, come to some conclusions, or, oh no. Okay, sorry about that, y'all. Hopefully you could hear me all right. But if y'all can hear me, put a 1 in the chat. But anyway, I spoke with AM1 in the comments. We were going back and forth. So I said, well, let's just do a stream about it. We can talk about this, hash everything out. If there's any issues, and go from there. I think it was mostly on the vaccine, maybe the development of the vaccine. I'm not sure. What's going on, Salchi, Leonard, MLR? Everybody hit the like button on your way in. Before I bring AM1 in, because he's here, I wanted to do some housekeeping. First of all, if y'all couldn't hear me earlier, my bad for that. I'll handle some little business. But thank you to all of you for the support. I'm going to try to answer. I know people have comments in the comment section. I miss them and whatnot. But I try my best to put a heart next to the comments that I get. Some of them I'm not going to put a heart by, because you know who you are. You know what you done did. But I usually put hearts by all people's comments. And like I always say, if you have questions, you can always email me. You can come in the stream. I'll send you the link. It's in the chat, whatever. And you can talk, on the stream. I'm not going to go back and forth with people in the chat. It's ridiculous unless you say something that is pertinent to the discussion. Alicia had mentioned something on one of the streams about herbal and natural remedies. So that's something I think she and I or other people can discuss, excuse me, at another time. But I thought it was important that she mentioned that, and I wanted to just shout her out for at least bringing up that topic in the, in the environment we're in with COVID and everything. What's going on, AM1?
Hey, what's up? Can you hear me okay?
Yeah, I can hear you.
Okay, cool. How you doing? How you been?
I'm doing well. How are you?
I am good, except for being hella sleepy right now because I had crazy sleep last night and the night before after like a crazy week last week of doing a whole bunch of client work. So I've been like, just doing a lot. Now just trying to recover before I get back into more work. But overall, I am great despite the craziness of this world, I guess.
Yeah, I feel you on that. I'm glad that you're here. Everyone go to AM1's channel. Type something in the chat. You can click on AM1's name on his icon and go to his channel and subscribe. He has a lot of great content. I did check out the video that you did with Erica Williams. I like the quality of the videography is what I look for, and it's there, and the subtitles. I checked the subtitles. Thank you for that. I appreciate that because they were outside and they're doing big things out there. She's in Austin, Texas, right?
Yeah, yeah, she's doing it big. She
Yeah.
she's she showed me a lot of stuff, taught me a lot of stuff. I actually just started a new business. I got a semi truck. I'm just soaking up game from her. It's another one of the things that's just on my plate, crazy. So even I told her earlier this year when she was kind of showing me the game on the whole trucking business. I'm like, wait a minute, what? And I was like, you know what? I'm getting one. And then here we are in July, pulled the trigger on it. So a whole new business on top of the regular work I do, my main work.
Mm-hmm.
Yeah, she's doing it big though. Yeah, the whole trucking stuff. She's serial entrepreneur. Y'all definitely go check out Erica Williams. She's super dope. Nicole is referring to a video piece that I emailed the link to her, Nicole, about Erica. I've been working with Erica for wow. It's been since last December. With the kind of stuff she does, serial entrepreneur. She goes to a lot of different workshops and just travels around the country, travels around the world. So she had me come out and fly to some spots with her and come check in with some of these networking events. So went to fly out with her and sort of capture like a reality show, TV show style day in the life type of just how she gets down. Here's how she networks, and it was super dope for me because she gets it in with a lot of other like Black entrepreneurs and not only Black, but she just introduced me to this network of just folks that's getting it in, and it's just really inspirational. So that's just one part of what I'm doing, but that's the piece that Nicole is referring to.
Mm-hmm. Yeah, I just put her link to her channel in the chat room. Thank you, MLR, for that cash app. Mm-hmm. So we'll keep it moving. I was doing some housekeeping, but I didn't want to leave you just sitting in there waiting. So I brought you up. But other people I wanted to shout out, Raining Woman. She has a great channel. Go check out her streams and everything that she does over there. I just like her energy is just very positive. So if you're looking for a positive channel, and she talks about a little bit of everything over there. I have a few clips on my channel where AM1 and myself and many other people were talking about Black plight versus procreation. And I thought it was great. A lot of people still email me about those streams and the clips that I took from the streams as well, just to bring people in, just to see that Black people talk about a lot of constructive things. And I think it's important to keep mentioning that without the effery on TV and whatnot. And even in the YouTube, Crimson Cure, Kendra, of course, y'all know she's on fire every day. And she's I thank her for that. She stays on top of everything that's going on as far as love when you need it. And that's what I appreciate about her because I'm going through, so I appreciate her channel. That's the Crimson Cure. And people think that I'm stuck in a cubby hole of science and tech, and that's just not the case. I a few things that I do, and today I'm bringing one. I would hope that we can have a constructive discussion about Koch's postulate and these vaccines, because you had sorry about that, Salchi. Hopefully it's getting clearer. Discuss any questions, comments, concerns that I'm having some little issues. No, I haven't dropped off. I'm right here. I haven't. I think.
you hear me okay? Or does it sound like it's Borgen? Hello? Hello?
You can hear me okay?
Oh, shit. Nicole, can you hear me? Nicole, can you hear me?
Try to join in from the other side.
Can you hear me? Nicole?
Yeah.
Nicole? Damn. Let me try.
All right, I didn't came
Other side. My bad. Let me mute this.
No, you good. I thought my internet was tripping. I was trying to reset my modem.
Okay, let me mute this. Hello?
Yeah.
You can hear me all right?
Yeah, it sounds cool. Now, it seemed like it was Borgen at first, and I thought that was my internet.
I'm looking like, what is going on? Like, it's ridiculous, but I'm like, what is this? I'm like, you're Borgen. Thank you, everybody, for putting a super chat in there. I appreciate it. I'm trying to figure this thing out.
Oh, they can hear it too? They can hear it too.
Yeah, they were telling me I was reading the chat while I was going. I'm sorry for that, everyone.
Okay.
You said your internet was, I see right here. You said my internet is tripping. Yeah, mine just started tripping all of a sudden. Like, what is going on? It was like acting crazy. The screen is doing something funny. Anyways, I'm just gonna have two of me up here, so we're gonna go from there. But yeah, what was I saying? My apologies to you all. Everyone let me know. BGS said white man holding you back. He hold both of us back. White man trying to hold me back, AM1.
White man don't want black folks having discussions like this about this particular topic.
I'm like, what is going on here? We talking about something, I guess, too serious for them.
Right.
But we talking about something that's like centuries old, you know what I'm saying? But it's cool. Right. I bet you if we talked about Cardi B or we talked about Sheree off Real Housewives, they'd have been like, oh yeah, go ahead and let them do their thing.
Right.
They wouldn't have bothered them. But anyway, we're gonna get back to what we were gonna be discussing. So I'll bring it back to the comment section of the video I posted, and you had comments in there, and I was interested, based on your other video, having patience with the patients, what you thought about the video, you know, if there's anything that, just the subject matter in general that you wanted to discuss.
Oh my God, I'm sorry, I'm so spaced out on it too. God, and the amount of videos I'm watching, I can't even remember exactly what it was about. I remember chatting in it, and I remember you sent me a link to a study that was done because I asked about Koch postulates, and I looked at that, and like I told you in the email, a lot of that went over my head, and then I know that you sent me like another graphic sort of thing, and it was kind of pinning down more of Koch postulates, but off the top of my head, I can't even remember the topic of the original video that I was commenting on.
So the video I had was on I did, because I've done so many with COVID lately. I think it was the one about was it hydroxychloroquine? I think it was the Oxford University study. And Brother Danubian made some comments in there. You made comments, and I thought they were interesting because it's, you know, there still are questions out there. People who are working in this field every day, like directly running these clinical trials, they have questions, I'm sure, as well. So it was just interesting to me that people who are not connected directly with that field have questions, and not just the question like, oh, is this caused by 5G or something, but questions that are more in depth. And, you know, you brought up something that nobody else really brought up that's in the field. People bring it up, but outside the field, it was like, okay, now I know, you know, who I'm working with now, you know, and that was good. That was interesting. So I appreciate that, because I put stuff up and I'm waiting for somebody to like ask something or say something. And then I'm like, okay, now, you know, here we go. Now we can have a real discussion.
Right. Okay, so I know exactly what you're talking about now. And yeah, so I now have a reference point. But yeah, no, I mean, since this whole thing started kind of rolling out, I mean, I'm not too trying to think of the right way to put this. I'm not too in favor of the idea of, well, you know what? I'll tell it straight up like it is. Because, you know, I'm a videographer. It's just so everybody knows, like, my whole background. I'm a videographer, motion graphic designer out of the Bay Area. So I'm working with a lot of different organizations, nonprofits, tech companies, financial, fintech type stuff. What am I trying to say? Venture capital. I have all kind of different clients. Erica Williams. I work with different YouTubers. I create content for content creators. I've been studying this stuff for years. I'm about to be 42 actually in a couple of days, but a good quarter, almost half of my life, been in the mix with this kind of visual media type stuff, from graphic design to motion graphics to music to events to just all kind of different stuff relative to multimedia. And I'll preface it that way because my whole stance on a lot of this stuff is just, it's relative to me looking at it through that lens. My work is all about capturing something and presenting it a certain way. Luckily, I'm never in a position where I have to deal with clients and spin something that is negative and make it seem good. In most cases, it's something entertaining or something enlightening or something that's of a positive nature. So it's just kind of a matter of, you know, just connecting the audience, connecting the viewer with what the client is trying to do, or that event, or that, you know, piece of information, that piece of content. So again, I preface it that way because everything that we're hearing about, you know, this current situation now, when I first heard about it, obviously it's through the news, it's different reports, social media. I don't really watch TV because I don't even have like cable like that. So the networks, but you know, the same networks that used to be just on TV are on even YouTube, or we're on YouTube now, or all over the internet. So seeing this stuff, oh, there's some kind of new virus thing popping up in China. People are getting sick, this, that, and the third. People are falling over. All the videos they were showing, I automatically was like, yeah, right, okay, because I don't trust mainstream media at all. Like I had a whole series on my channel. Mainstream media is not your friend. So I mean that has just been the case. So, and then we want to get into governments doing all kind of shady stuff, classified stuff, black ops type stuff, psychological operation type stuff, especially relative to, you know, black folks would get to the Tuskegee thing. So experimentation, there's history of that all around the world, different, you know, non-government organizations doing all kind of clandestine type stuff all around the world, doing all kind of funny business. Bill and Melinda Gates Foundation, just different stuff. So I'm looking at it like, oh, okay, they're talking virus in China. I'm like, all right, here we go. I figured it was gonna roll out to something like what we're seeing now, but it was amazing to just see it roll out. Like, wow, we start getting the reports of, you know, it's showing up here, and then now it's spreading. And cruise ship, they're gonna bring the cruise ship to San Francisco. Oh no, they're gonna bring it to Oakland. So just, you know what I mean? Like just looking at it. So the mainstream media, we're hearing it from now the U.S. government. We're hearing information that's surrounding a virus. So then, of course, we're getting into the medical industry. We're getting into the pharmaceutical industry. So I call that the big four. Those four are all known corrupt organizations that have had all kind of different issues, crimes against humanity level type stuff, deception, it been exposed, all kind of different scandals and whatnot. So, I mean, this is all stuff we all know. So for me, hearing how this thing is rolled out, I'm like, let me start asking some more questions about this. Let me start looking more into this because I'm not buying it. And I want to know more. They're telling me there's some kind of thing that can infect my body in such a way. And of course, I've heard that my whole life. I'm like, let me look more into that because I'm into health. I'm into health and wellness, fitness. Again, I'm about to be 42. I'm trying to stay up on my health. And it's empowering. Plus, since I'm a freelancer, I can't get sick. I got to be on top of my game. Plus, who wants to get sick? Let me learn more about this. Let me look more into virology. Let me look more into the whole nature of this. Oh, so virology, how that kind of dates back. So we're talking 150 years. Louis Pasteur, we're talking Robert Koch, who's some of the other folks too. some of the
Yeah,
names
it goes
are
back
stupid.
to Watson and Crick, and yeah, a lot of people DNA and because viruses, DNA, genetics, and how that can affect you, and introns, understanding introns and exons, all of that goes into it. I understand that people are skeptical, and that was one thing that the skepticals, the detractors, people who are or not, maybe like not from what you're saying, you're taking it seriously, but you're looking at it holistically. A lot of people don't do that, and I know that. You're obviously saying earlier, you were talking about what you said before, and you stick to that. And I agree, mainstream media is not your friend. I literally saw the other day, I think it was MSNBC or CNN, but there was a school in New York where they're saying, we have the kids coming and we haven't had any COVID infections, but they're testing their temperature with a temporal scanner, which is not used anymore, really. It's not as accurate. So when you are testing someone's temperature on their forehead, and I know Amazon's selling them out the wazoo, but they're kind of useless, and well, not really useless, but more obsolete than anything right now because they're not as accurate. So the way that you're supposed to test is behind the ear, behind the left or right ear, it doesn't matter, but it's not on the forehead. And I saw that and I was like, hold up, how are you going you're saying that you don't have any infections, but you're testing the kids' temperature when they come in. Some of the kids' masks weren't even over their noses. And I was like, how are you going to keep, second graders and third graders from infecting each other? Or what is the testing schedule? They didn't talk about that. It was just very sketchy. And so I said, well, that's not something I believe. This is BS. And there are a lot of things like people, I don't never assume that I'm believing everything I'm seeing on TV. The reason why I put these videos up is to make you think, look at what they are writing, look at what someone got paid to write and put in here. What does this mean? When I tell you click the links, you see the articles, because the person writing is paid to write a certain way for that publication, right? But the real information is a lot of times in the blue underlined highlighted links. This study from Nature Communications last week said this, and then they go on and talk about something else. Click on the link and see what they actually said, see what the scientists said, because the journalist is not going to put the statistics. They're not going to put in the article where they said, oh, this experiment didn't work as well, so we did this other experiment. They're going to just say what worked, because that's their job, And, like I said, I was on an art stream just a few minutes ago. It was like, science is just, anything is a science. It's just, to me, it's just an intense study. You take the time to go through the scientific method, observe a problem, develop a question to answer the problem. And then you go through experimental methods. You design methods if you have to from scratch. But if you don't want to, you can take methods that someone else has developed and published, devise your own method or use theirs to answer a new question. Once you answer that question, you conclude what happened. Did it work? Did it not work? And then you make an inference to develop your next study. That's just basic scientific method. If the logic does not, if you can't get past the observation to try to develop a hypothesis for why, if this is a NGO or even a governmental plot or plan, but you can't get to the point where you're developing what their hypothesis could be and how they're developing their strategy for population control or whatever that may be, then it falls flat. The logic falls flat. That's just how I see it, because anything you can pretty much use a scientific method, and just start from the beginning and work your way down to see, does this make sense? And that's how Koch's postulates pretty much was developed. You need to observe his postulates are making the observation over and over. Basically, that's what it is.
It's like a process of elimination, right?
Exactly, process of elimination, and observing something happen in two different quote unquote universes, like in one mouse versus another mouse, or a mouse to a rabbit, or a mouse to a frog, or something like that, which you can do. From a human to a zebrafish, which you can do.
Interspecies infection.
Yes. Can you infect in between species? Can you affect within the species, inter or intra species infection? Can you quantitate the infection that's coming out of that species? All of that makes a difference. And Sachi is right. It's a discipline. You know, what's going on, everybody in the chat? I'm sorry, I'm not looking at the chat right now, but hopefully you're enjoying the discussion that we're having. What I'm going to do is try to share my screen here, since we kind of moved into Koch. I'm not sure how to pronounce that. I don't know. Can you all see this screen? Koch. Okay, hopefully it's coming up for you. All right. So this is from microbenotes.com, and I literally just typed in Koch's postulates, and this was one of the things that came up. So there's a table of contents. This is pretty long.
Wait, can everybody, can the viewer see your screen? I see it in StreamYard, but I'm like trying to understand. Can they see it?
Hopefully they can see it, because I can see it on my see it on my phone and on my computer, you know, like 3 different ways.
Yep.
Yeah,
okay,
so
yeah,
they have a table of contents. This was published last year, January 29th, 2019, which I think is interesting. And updated January 10th, 2020 by the same person. So they go through, you had a man named Heinrich Hermann Robert Koch, or Koch, and he is just credited as one of the founders of the specific field of modern bacteriology. So his basic principles were for tuberculosis, cholera, and anthrax. And he wanted to give experimental support, which he did, for the concepts of infectious diseases, which included experiments on humans and animals, excuse me. And for that, he's regarded as a pioneer of public health. Because at the time, this was, let's go back up to the years, 1843 to 1910 is when he lived. So yeah, there was a lot of diseases and infections going on at that time. Now, another thing is what he did aided in legislation and changing prevailing attitudes about hygiene to prevent the spread of various infectious diseases. That's very important, because it shows you that science, as I've said before, science does affect policy. The development of certain technologies affects policy. So for his work on tuberculosis, he was awarded the Nobel Prize in 1905 in physiology or medicine. So Koch's or Koch's postulates, I'm gonna say Koch. I think because I'm saying because of the Koch brothers, I think that's why I'm saying it like that. But anyway, he has 4 different statements within the postulates. Number 1 is the microorganism must be found in abundance in all organisms suffering from the disease, but should not be found in healthy organisms. And so we can check that off the list, because that, if you have it, if you have COVID, you have COVID. And whether you have symptoms or not,
wait, you said if you have COVID, you have COVID.
If you have COVID, you have it, whether you have it with symptoms or not, you're infected. And there have been a lot of people who have symptoms, and there are a lot of people who do not have symptoms.
Like that.
So you're suffering from disease. Suffering is a carrier. You could be a person with sickle cell and have less aggressive form of sickle cell allele. And you know, physically you can't really tell this person has sickle cell, but they have it. They just are not expressing the phenotype at that time. Then you have someone else with more severe sickle cell, where it's very obvious, you know, in their physiology, you know, phenotype is showing. Then you have others who are carriers who will then pass it down. So with this virus, it's not the same as sickle cell, but you have it. You're carrying this virus around. It's just a matter of time, or a matter of it affecting your immune system to the point where you potentially will have symptoms, or transmit it to someone else, you know, through water droplets, and they will have symptoms, you know. So it's a 50-50 chance for that, but it's not in healthy organisms. And so the next part they say is the microorganism must be isolated from a diseased organism and grown in pure culture. So this is usually where a lot of the studies fall flat, and it can't go on to the rest of the the other two. So this is where the study that I sent you, this is proven, where the microorganism or COVID must be isolated from a diseased organism and grown in pure culture. And that happened. They did that successfully. The article is supposed to be published August 6th, but it was published early online. I think it was late June or something. So I'll show that in a minute. Then the third thing is the cultured virus should cause disease when introduced into a healthy organism. And they did that in the study because they were able to collect monoclonal antibodies from the second mouse and detect them in their blood. And the last thing is the microorganism or COVID, the virus, must be re-isolated from the inoculated diseased experimental host and identified as being identical to the original specific causative agent. Yes, they did that up here, actually. This part it wasn't done in order, the study that I sent you, AM1. But they did 1 and 4 together, and then 2 and 3 after. So the study I sent you, what I posted in the comment section, was a 3-step process that was, as you saw, it was a very long article. But hopefully you can still hear me.
I hear you.
Okay. So that everything else just goes down and talks about what he was interested in, like very specific things. Let me move it down some. And basic preparation of samples, things like that, type of things. Oops, let me go back. It went on to something else. I clicked something else. So they go on to his major contributions, and that's okay. It's all right. It's cool. But your main thing was here, and I don't know. I mean, to me, they have done what's in here. Maybe I'm missing something. Let me try to make this a little bigger.
I mean, I was asking about it relative to some articles I was reading and some videos I was hearing, some presentations I was hearing from some different doctors, MD, microbiologists. There's a few of them that I was kind of checking out, and they were talking about how they have colleagues that they were passing around some of these different studies and these reports as this whole COVID thing has kind of been bouncing around, and that they're, reading through these things, and they're sort of, studied in how to sort through this stuff. Like with the study you sent me, and I outright said a lot of this stuff goes over my head, but I was kind of able to parse through it a little bit. But these other folks I was kind of checking out, like, I'm not necessarily saying what they're saying is true, but they're raising some interesting questions that parallel with some other books that I was reading. And I just, over the past few months, built up a very interesting collection of kind of old books talking specifically about germ theory versus terrain theory. So the whole Louis Pasteur, Robert, or what's his name? There's Robert Koch, and then there's the other dude, Pierre Beauchamp. I'm looking at the history to now and kind of the evolution of what should be the science, like, the scientific method, how you broke it down before, and the idea that as new technology progresses, certain things should be able to be looked back upon and built upon and innovated upon. And it seems that they might not be doing that. But these doctors were outright saying that the COVID or the SARS-CoV-2 virus that causes the COVID-19 disease relative to the symptoms that they cause, or that the virus causes, and that ever expanding symptoms list, which is interesting because it's similar to HIV and AIDS, how they just kept adding on symptoms. Basically, they're outright saying that it has not been isolated by way of Koch's postulates, that they haven't actually shown it to have worked this way. These doctors are claiming that pretty much most of the major viruses that we've known in the past 50 years haven't met Koch's postulates. So that just collapses a lot of the whole concept of, one, what is actually making people ill, and then obviously the subsequent idea of being able to isolate said virus and create a vaccine out of it. So that creates some very murky sort of discussions. But I mean, these people, you know, I don't know everything they know, but I'm just, you know, listening to it in the sense that I have these sort of like virgin ears to hear this stuff, and I'm not trained in any sort of way, and I don't have any kind of like incentive. I'm not indoctrinated into that in any sort of way. So I get to look at it with this clean slate, like, huh, okay, interesting. Yeah. But then I have to look at it from the standpoint of, are there potential motivations as to why people would sort of push this stuff through? Can studies be bought? Which I know is true, because now we're getting into that. We're talking about those big four known corrupt organizations who we've known and who are notorious for doing all types of shady shit. So
yeah, I mean, BGS mentioned something, you know, I mean, isolating it, first of all, you have to remember that a virus can be like 1 nanometer,
right.
0.5 nanometer, which is 1 times 10 to the -9 meters small.
Right.
So you're talking about nanoparticles. Now we can see nanoparticles, which I can show in a minute. Nanoparticles under microscope. I have gold nanoparticles in my lab in the fridge, 4-degree fridge right now. It's gold. It's 24 karat gold nanoparticles, gold that has been pulverized into a nanoparticle and then run through a sieve to only collect the rounded, you know, the ones that are a perfect sphere. And those are the gold nanoparticles I use for cysteine isolation from blood. When I want to take the cysteine from blood, cysteine being an amino acid, and I only want to analyze that amino acid, then I get all the amino acids, and then after they attach to gold, I go and you run them through another process where the charge pulls off cysteine because it has a specific charge. And then I only look at cysteine on a mass spec or something like that, you know, some type of expensive equipment that I'm just learning how to use right now. But I know what the gold is, you know, gold is inert, gold is platinum. These things.
I was going to say, are these actual living biological material?
It's blood from a human.
Okay.
Patient blood that I take. These patients are dead, but we have their blood, and we want to see if the drug we gave them affected their blood amino acid content, and certain amino acids can be used to develop medical devices. So that's what we were doing, and that's just the initial stages. They're not going to put gold plate, 24 karat gold plating or platinum plating on a medical device. It's too expensive. But in doing the initial stages of the experiments, you need something that you can reproduce. You can use as reproducibility, increasing your statistics and whatnot. But this is just a cartoon image of what they saw on the electron microscope of a virus. This is probably a T4 phage. So this is a type of virus that does not infect humans. It only affects bacteria. That is its only job. And you can program it in the capsid head with whatever, put whatever DNA, RNA you want in there. And once it gets to the, the bacterial culture, it pushes its genetic material through. This would be, I guess, the picture cartoon image of what the plasma membrane would look like, or the cell wall. And then it would push its DNA or RNA into that cell, infect it, and then move on to another cell. But if you have thousands of these, then you have a problem.
I don't want to cut you off, but I want to ask you about this, because kind of relative to these presentations I was hearing from these speakers, like this slide that's at the top in the middle, this black and white slide, they were saying something to the effect that even if they can use an electron microscope to look at biological material that's that small, that ultimately the process for looking at it through an electron microscope requires for certain chemicals to be used, and that there's like these real small or super thin like cross sections to where it can even work on a microscope, and ultimately it's dead material, so it's not even living.
Well, the identification,
because I guess the chemical process would kill the virus that they're trying to even look at, and the process for looking at it through a electron microscope kills whatever biological material. Is that?
Yeah, well, that's true for most things you look at on a slide. Most things you look at on a slide are dead, unless you're doing a live, live camera.
Oh, it's on a slide. Okay.
Like if we do, if we want to look at migration.
Damn, it's chopping up. I'm like, is that just my signal? It's borging on my signal, or I can't tell if that's you or just my
No,
signal?
you
Damn.
sound fine for me. You sound fine. I can hear you fine.
Damn.
I can hear
Might
you
have to
just
switch to my
fine.
phone. Maybe my internet is tripping.
I had to switch to my phone earlier. I don't know what they're doing. Like,
Yeah.
it's crazy, but I'll just keep going. But most things that you see on the slides are dead. Even tissue, human tissue that's being analyzed with some type of antibody, that's dead. You can't do that analysis without the tissue being dead, without the animal that the tumor came out of being dead. So that you can stain the tissue and see if there's a change in protein expression based on some drug that you gave the mouse, right? And that will give you an idea of what happens. That gives you an idea what happens in a human if they're given that same drug. So these are just images. These are not even like, I put retroviruses, as you can see in the top here, but they haven't given me retroviruses. They've given me more T4 phages. This is just the name, T as in Tom, 4, the number 4. And as you can see, this is a scale. This line, anything this long is 100 nanometers. So this is a big virus that's supposed to be infecting bacteria. And they have to be bigger because you want a high infection rate for your bacteria. So maybe this virus has some kind of gene in it that you want the bacteria to carry. And so that's an easy way to doing it without like having not nowadays you can use CRISPR, you can use all these different things, but back then that's what they used. And they do like radiographs to determine if the expression was there in the bacteria, because they're eventually going to die, right? So you
If we're looking at
bacteria and
slides
then look at the DNA.
of,
So everything here is
we're looking
dead.
You said all those things are dead, and they use like a chemical process to make the slide. So I'm just curious, like even within, because I was reading about this in this book called Goodbye Germ Theory, and some of the other presentations from these doctors, they were saying that and again, I don't know, they're saying it can't have met Koch's postulates just alone on the premise that, one, you have to really be able to isolate exactly what you're looking at, but you need to know that it's actually alive, and then you need to be able to remove it and culture it and then reinfect another organism and actually be able to perceive each of those steps with a living virus particle. So it almost seems like straight away, if the only way you can even look at it is for it to be dead, it seems like that kind of, it's not even alive.
I won't say the only way that, because BGS made a good point. He said, I think he said being alive is relative. It is a relative term. And when it comes to viruses, you have to look at the effect of the viruses, and then you have to determine how many copies or colonies of the viruses, of the virus that you have, is being produced. So with CRISPR, and what's going on, Anne, she says, seems to me like they are making viruses with CRISPR. So you don't necessarily make a virus with CRISPR. You start off with a vector backbone, and then you add in whatever you want, whatever genetic sequence you want. You put stop codon target sequences in there. If you want to cut a gene out, you can put that in there, and the cell will no longer make that gene forever. You can put a new gene in that the cell never had before. You can switch genes around and cause a shift. Like if you switch 2 genes around, you reverse them, then their sequence is backwards, and eventually that gene will be cut out because it's an error. It's a major error, right? And you can test for that. So if you did that with a virus, like you have to use some type of viral agent in order to do that. And the way that you determine that it's there, you have primers to do your PCR for the sequence, for that viral sequence that's foreign, and you have primers for when it's knocked out. So you can determine if the virus is there and the virus is not there. You have to set up your controls. And so the paper that I put in the comment section, they had like 3 controls for their experiment, which is that's beyond like basic science. You don't really have that many controls in a basic science type of situation, but this is translational research, but it's still based in basic science because they're using like adenoviruses, which are more than 30 years old. The type of technology is not new. It's very old. But she said, viruses are essentially parasites too, but scientists are not addressing it as such. Of course, yes, they are parasites. You know, it's just a matter of whether you're, to me, they're parasites, but you can have a parasite in your body for a long time and not know it's there. It's based on your immune suppression or immune activation. What level of immune suppression or immune activation are you in? You know, whether or not your immune system is going to say, hey, something's wrong, we need to do something about this. However, viruses have the ability to mutate. So once your immune system realizes there's a problem, there's somebody here who shouldn't be there, they've already taken off their blue jacket and put on a red jacket. So your immune system cannot recognize that virus anymore. It's a brand new problem, right? And by that time, the virus has gone from your lungs to your heart, or from your heart to your liver, or from your liver to your brain. So you're stuck. And in trying to treat that, you may actually do more damage than actually helping the patient, because you're treating them with the initial infection and not the current infection, which could be 2 or 3 organs. You see what I'm saying? So with viruses, to me, it's a parasitic infection, because you have sometimes similar symptoms as you would with the parasite. And some of these viruses kind of look like parasites, you know, in a way. Ebola is the one that comes to mind, that somewhat looks like a parasite or a worm. The destruction of the organs, the destruction of the bloodstream, the circulatory, lymphatic system, that's a lot of times what you see with parasitic infections. But viruses also are so easily mutable when they're like this RNA virus. And now that's the difference right there, the main difference. They're smaller than standard worms or parasites or helminth. A helminth is just a worm, roundworm, flatworms, things like that, and easily transmissible between person to person. You know, the vector is not as clearly recognized with the virus as it is with a parasite. Parasite might be bad water, bad soil or something, or bad food or bad milk or something like that. But with the virus, I mean, person to person contact, you know, breathing around a person, you get it.
Well, I guess, and I've heard actually a lot of what you're describing, but from kind of the information that was kind of piquing my interest, it made me kind of take it back a step further to sort of the initial discussion. And this is Pasteur versus sort of Beauchamp, that had these kind of different outlooks. Again, the germ theory versus terrain theory concept, in that what people were sort of identifying as virus particles, these inert non-living things that sort of latch on to cells, insert their RNA and sort of hijack those cells and then replicate themselves and then potentially have these pathologies. Is some of the people are saying that that's actually not the case, and that what's happening is some type of chemical or outside thing is happening to your body, or it could even be foods you're eating that is harmful or destructive to your body, and that it's actually the poor terrain, meaning you're not keeping your health up and your cells are deteriorating, and the deterioration of the cells in the form of what is this word, exosomes, or even Beauchamp talked about microzyma, and I did in this book, Goodbye Germ Theory, basically saying, questioning the whole concept of germ theory, talking about terrain theory and the idea of like not keeping your gut bacteria right, or not sleeping right, or contacting some type of industrial chemicals or that are causing certain destruction in your cells, and people looking at that and saying, yeah, see, they're having certain symptoms. What we're seeing in these microscopic cells, that's the virus. And again, that's why I found it interesting with the whole idea of the electron microscope. They're not looking at something that's alive. So that does seem like it leaves it open for them to kind of label it however they want and say, this is the thing that's making you sick. So it's the whole idea of like causality. Can they prove the actual causality? I don't know. I mean, again, I don't know this stuff. So this is me just trying to parse it out and piece it together myself, because as I'm looking at it more in some of what these doctors are saying, there's an outright motivation for how they set it up this way. And it's to the tune of billions of dollars, potentially trillions of dollars over the last century, that the science may not be quite what we've told this whole time. I don't know.
Yeah.
Have you ever heard about that? Like the whole idea of germ theory versus terrain theory?
Yeah, I put a link to that. Or
I should ask you that
not
out of the
link,
gate,
but the
actually.
yeah, you did. I remember I put the information for that. I couldn't find the link, but I think I might have put the link later, but I put something about, because you asked, I think you and Brother Danubian asked about the exosomes and the germ theory, and I haven't read through that in a long time, but I did find this. This is an article that is on NCBI. So if you go on NCBI and you type in this number that's here at the top, 224, and then you can actually just type in this 224 number you can type in, or you can type in when is a virus an exosome, the title, whichever is easier for you. Just put this in Google. This should come up for you. And this is the research roundup. This was published in 2003. There's actually 2 articles here, but it was 2003 when they were talking about this, and they have it referenced. This is the Proceedings of National Academies of Science, the USA, and they have just 2 references. So Dr. Hildreth, James Hildreth, I've met him actually years ago, and very intelligent man, Black scientist, and very important information that he gave at that conference. He's from Johns Hopkins, and in Baltimore, and they talked about here, this is a Boldness Theory, where Dr. Hildreth actually stated, and they quoted him as saying, hold on, let me come down a little bit. He proposes at this point when this was published, he proposed that the virus, they were talking about HIV, is fully an exosome in every sense of the word. And what he meant by that was, and Gould, this other person, Stephen Gould, also agrees, and he said that HIV is an exosome. It's not just a protein, not just proteins in a vesicle, but it's something that is meant to be trafficked from cell to cell. And so that's what a lot of people were having issues with, trying to determine what the virus was. And if it's a vesicle and it's something used for trafficking, then it's something that you can detect and identify. And so there was a lot of questions at that time. There still is questions about, like you were asking earlier, can a virus be, can you question whether a virus can be identified? Can you see it? Can you get the concentration of it in your blood? Can you get the concentration of the virus in serum, or can you reproduce this? Basically answering the postulates that we were talking about earlier. That's really what they were talking about in this article.
It just dawned on me. I have to see this guy's presentation, because he's breaking that down, like relative to what you just said, being able to see that. And I think the phrase for that is viral load. So he made the comparison that what they're describing as a viral load is actually exosomes that are potentially being kicked off by your cells because of some type of chemical insult or injury. Not necessarily that it's something that was some exogenous thing that entered your body and that was inert in some sort of way, enters your body and comes alive and then starts hijacking your cells. Basically, he's postulating that the whole concept, again, that germ theory isn't what it is, and that it is more of a terrain theory concept, and it's something that we're essentially doing to ourselves, and that we ultimately could heal ourselves if we understood it properly, which then obviously opens the door for why more research wouldn't be put there, because then you can't profit from it. You see what I mean?
Yeah.
That's kind of where they were going with that. But
I'm listening, because the profiting is important, because how does science get done? You have to have money to get science done. You have to have money to get things done. I just wrote a grant in May to get, hopefully I get it, I don't know, but I wrote a grant and submitted it back in May to get money to continue my second project. And I'm still finishing up my first project for publication, but the work has to be funded some way. We can't, a lot of the stuff we buy is very expensive. I have certain viruses that I want to design from scratch, and I need help, and money, to do that. And you can have the best idea in the world, but if there's no money behind you, it just sits still, like anything, it sits still and never goes anywhere. And in science, we don't work in a vacuum. Things are funded. We collaborate with a lot of people. They have to be paid. They got to eat. And if you go and you have a idea that you want to present If you want to submit a patent, that costs money. I mean, that might be up to over $100,000 just to get a patent off the ground. You know, just to submit the initial applications and all the documentation that needs to go into that. That's, I mean, if you ever looked at a patent online, it's a lot of documentation on there. You know, not just
I've seen it.
pages. It's a lot of figures, and you know, I mean, it could be up to 300 pages long. You know, and that takes money. Someone has to allocate resources for you. Someone has to get you licensed. Someone has to go to the government and say, you know, hey, this is a great idea. We got an idea for a new medical device, or we want to update a previous medical device that we developed. Well, you can't over trademark yourself. You have to trademark again, or, you know, patent again. You know, you can't plagiarize yourself, right? Because a patent is a publication. So, I mean, everything is dealing with money. But I'd like to see, you know, more of the money go to things that, I don't know. I have a lot of ideas, but I have a lot of things that I would like to see that go towards, like, I don't know, more development of technology. And I know they're doing that, but in this country, you know, other countries do it. Clearly we see that. But yeah,
that's an interesting point though, because some of those doctors were talking about the idea of that exact point, like certain labs, certain independent researchers trying to obviously be funded, and that could be to the tunes of, you know, millions of dollars that could hold down their lab for a while, depending on, you know, the kind of research they're doing. And they specifically said, well, yeah, a lot of things that could result in actual healing technologies aren't often funded, or they'll be funded and then essentially disappeared, or they'll just get rid of that, because if they're like, well, if you could cure something that we could actually profiteer from over time indefinitely, then of course we'd rather do that, because that's a better business model. So they're saying like that alone introduces the potentiality for corruption. So for me, as I'm looking at it again from a naive sort of mind state, just a blank slate, and what I understand about media, governments, pharmaceutical industry, known corrupt organizations, it stands to reason that, maybe they did force this germ theory concept on us, when maybe there is some more natural thing. Like obviously our bodies are super interesting and some amazing kind of thing that we all, you know, live in, right? So maybe they got it wrong, but on purpose, and there is some other way to describe it, as again, the germ theory versus terrain theory. BGS asked a great question: how do people transmit these from person to person? I guess he's asking that relative to what I was saying these doctors were proposing. If it is something like a terrain theory, or, you know, exosomes are being misidentified or classified as viruses, then there's the idea of bacterial infections. Like germ theory would be microbes, right? So we're talking about bacteria, we're talking about parasites, we're talking about viruses, we're talking about fungus, all these things that can get down into these like microscopic particle levels, and that could theoretically enter your body, or already in your body, that are already a part of your body. And the idea of like cells that can sort of transform, I think the word for it is like pleomorphic cells, based on your body's needs or chemical makeup at that time. You have a certain injury, the cells will know how to transform and go address that particular issue. If you have a certain injury, they know how to clean up the cells. It's like autophagy, which I learned about from fasting.
Autophagy is something that happens too. Right. But I think when it comes to transmitting between person to person, so that's one thing where, because like I said, when I was talking about the exosomes earlier, that was something Dr. Hildreth and Dr. Gould were discussing during HIV, during the beginning stages of HIV, and also moving to immunizations and vaccinations and potential development of those technologies, or even treatments. They ended up with treatment, as we all know, with AZT and whatnot, and other treatments that they've developed since then, you know, different iterations of that. But when you talk about the exosomes are something that are, for me, that I have personally seen under the microscope, are little vesicles or little micelles that are extruded from the cell. They excrete these vesicles out, and we used to think that was waste. When we look in the microscope of a cell, they're very small dots. You can't see them that well, but when you use a high-powered microscope with higher objective lens, you can see that it's actually particles that little like cells, like little circles full of something, some kind of packaging communication type material that's being trafficked between cells during stress, or, you know, that's their, like we call them speech bubbles. That's what we term them as, because they're the way, that's the way the cells are talking. Of course, they use other ways of transmitting information to each other, but that's another way. If one cell is sick, another cell can send, that cell will send a message to another cell, perhaps to let them know, hey, I'm not doing too well over here, you might want to leave. These cells can move. They have pseudopodia and lamellipodia where they can move from place to place if they are at that, you know, aggressive level of cancer, for instance. So if cancer cells can do this, and then your regular cells can do this, because cancer cells are only transformed from normal cells. So the normal cells that you have in your body are giving off these exosomes anyway, and you get a viral infection, it would make sense that the viral bodies would be transported out. How do they get from being transported out between lung cells out of your mouth? Well, you breathe them out. You're coughing, you're sneezing. They're in the form of water droplets. You know, there's a certain timing where these vesicles or exosomes actually burst. You know, when these cells, like cancer cells, for instance, transmit them from place to place. When they go from one cell to the other, they get inside and they burst, you know, and then whatever material is there gets in that cell and it uses it for whatever it needs to use it for. So that could be ions, you know, whatever they're transporting back and forth. We're still trying to figure out what that is. A lot of it are small pieces of peptides that they're passing back and forth to each other. You know, like passing notes and stuff in class, that type of thing.
Right. It's a communication delivery system.
Yeah, so we see it as a communication device.
A heart cell can release certain exosomes from those cells because they need to alert some other part of your body, so that the exosomes are kind of part of a transmission system. No, I've read about that too. That was interesting. I'm just saying that these guys were postulating that Dr. Andy Kaufman and Robert O. Young, and then there's some other doctors that were kind of, I guess, said 30 years they've been looking at this whole exosome thing, but just saying that, understanding what exosomes can be used for, but just the idea that it's possible that way back when they first started looking for the causality of these illnesses, came across exosomes and said, bingo, that's a virus, and that they've basically mislabeled it. That's what they were saying. So I found that to be an interesting idea, and I see why institution-wise somebody would say, no, we want to say that's a virus, because then you can get into the realms of saying that's the causal issue of a particular symptom, while not addressing the real symptom. But now you can say, well, hey, here's a way I can maybe cure those symptoms for you in the form of a vaccine. Like I'm stating it in such a way where I'm trying to be careful to not make it sound like it's some type of a conspiracy or a racket kind of situation, but these doctors outright say that.
No, I don't see it like that. I just see that you're trying to gain more information and trying to, you know, get more into, I'm gonna use Art Nuss style. I'm gonna use his phrase. You're trying to gain more understanding, and there's nothing wrong with that. You know, people, like I said on Dr. Dunbar's video when I was with him, like, you know, I don't have an issue with conspiracy theorists. The issue I have really is like what BGS has said. You know, you don't have a, like, what is the end game? If you don't have an end game, then okay. If you haven't really thought out what's going on, then I don't really understand where you're going. You know, you have to have some type of train of thought, and it seems that you're developing that, and that's good. You know, I don't consider that conspiracy theory. Everybody has a theory. I have a theory every day how I'm gonna get to work in an hour and a half, like with all this traffic. And you know, that's just a theory. Like, how am I gonna do this? Like, let me estimate my timing and get myself together and get through this and figure it out. Get through this experiment, and I get to the end on time. You know, I'm here. And that's my conclusion. I got here on time. So with a theory, there's nothing wrong with that. The conspiracy comes where it's like, it's just outlandish. And okay, when has this, and then you have to look at, when has this happened before? When have people discussed this before? You know, that's how I see it. Like, I don't have problems with it, with the conspiracy theories, except they, like I said, it's outlandish, and they don't have a final, you know, solution. What's the solution that they're trying to come up with? And all this, you know, have you been reading? Have you been studying? And I think you have. From what you're telling me, you could definitely have. And that's a good thing, because you're not even in the field. You got people out here who aren't in the field and aren't taking the time to even see like this is something that's affecting us that will continue to affect us for a long time. And I appreciate that. You know, I
Yeah,
wanted
no,
to
I
ask,
mean, my thought process is kind of like, all right, well, wait a minute. What we're dealing with right now, when you kind of boil it all down, is certain people who are the quote unquote experts are telling you things about your body that you don't know.
mhm,
And I get it. There's certain levels of research or certain resources that you need to have, certain equipment you need to have to understand these things. But for the most part, if anyone can figure it out, I'm a smart guy. I can figure it out. So let me start trying to look at it to the best of my ability so I can understand how my body works. Apparently, I've been living with this thing for 40 plus years
right,
and will maybe keep living with it for God knows how long. But I'm concerned with the idea that can somebody weaponize my lack of information about my own body organism against me and start to tell me things and trick me into doing certain things like eating fast food or eating this, that, and the third, or GMOs and all this. And is it possible that somebody would tell me to consume or ingest certain things that could make me sick for the purposes of profiteering from that? Is that possible? Sure. We already know that's possible. We already know that's happening. So I'm like, why wouldn't everybody want to learn more about this stuff? I'm curious or mind boggled at how willing some people are to say, no, just go with what the experts are telling you. Don't even look at it for yourself. That's actually scary to me because you multiply that by millions of people, and then we arrive at a situation where somebody can tell you pretty much anything, and then people are fearful. People are thinking that there's some kind of virus situation flying around through the air, but they can't verify it or prove it themselves. And I keep asking people that, like, well, how do you know this virus is doing this, that, and the third? How do you know it mutated? How do you personally know that? Can
well,
you
because of
objectively prove that?
the
And
previous
everybody says
studies.
no, I can't, but it's because the experts told me. I'm like, well, by that logic, a so-called expert can tell you anything and you'll believe it then,
Yeah, from the previous
right? I mean,
outbreak, they didn't have many people die, but that was within the family of coronavirus, and that was SARS-1. And it was really bad I believe in China as well, many areas of Asia, because of traveling, and those countries are pretty close together. So they did have, I think it was like over 8,000 people to die from that. Um, and so they learned their lesson from that. And I actually think it was more people that died. It's just that was what they felt comfortable or they were willing to put out there as the death toll. And that's fine. A death toll is a death toll. It when it gets over a certain number, it's like just stop it, get it under control. So they learn from that, and there's several papers, several publications on the sequence of SARS-1, of the sequence of SARS-2, and how you can reproduce this in a mouse. And I mean, the study I have up right now, that's what it's doing, and what it did. And so this article, because you were asking about a lot of things, and I think this paper, and there are others too that are coming out that are discussing how you can like neutralize the or detect the virus, and the mice are being protected by the neutralizing antibodies that are being produced. So neutralizing antibody is one that should be produced by an animal that has a working immune system, or even an animal, even if the animal is immunocompromised, you'll have certain antibodies that are produced from the body that they can use at a high level to fight that foreign agent. And so we all know, or if you don't know, we know now that mice do not have the ACE2 receptor. The ACE2 receptor is what SARS-2 binds to on the outside of the cell in order to gain access into that cell. If it does not have an ACE2 receptor, how do you get ACE2? Well, you take a virus, adenovirus, that's what the adenovirus, that has the human ACE2 sequence on that. And they just given 5' to 3' DNA sequence. You put that into a viral package, and then you put that into the mouse through intranasal injection. So aerosol injection. 5 days later, you infect them with SARS-CoV-2 protein. And then after that, they have the infection in the lungs, and specifically they said the alveoli. So what they're doing is, as they say here, just understanding the COVID-19 disease, and they want to test therapeutics in different vaccines. So if it's in the mouse, that mouse produces antibodies. You take those antibodies, isolate them, inject them into another mouse, and that mouse will have the infection. So that's answering a couple of the postulates we talked about earlier. And that also cannot be a mouse that that will infect a mouse that already has it. This mouse has it, so this mouse is not able to reinfect this mouse. This mouse can go on to infect a healthy mouse. So that's what this line and this red line means. This means no, inhibition. So you cannot, this mouse infected with this antibody cannot go infect this mouse that has it directly, has the virus directly. Okay.
But again, this is based on it being isolated by Koch's postulates, in the sense that it can be isolated from the infected organism, be isolated, purified, grown in its own culture, and then reinfect another living organism. In our case, it'll be a human, though.
You can't isolate ACE2 from mouse. It's not there. They don't express ACE2. So it's the perfect organism to test this in, because they don't express the gene at all. So what they did was, it says they transduced these mice with adenoviruses encoding human ACE2. So they have a human gene in a mouse. That's what they did. So the human sequence for ACE2, which would be represented by this little cartoon, 5' to 3', that's a DNA sequence for ACE2 gene. The ACE2 gene making up the ACE2 receptor. So it is, what is it like? They're putting ACE2 receptor gene, the machinery that's needed to make that receptor is in the mice, and they didn't have it before. So that right there is already amazing that they could
they use genetically modified mice to prove Koch's postulates for a supposed human virus with GMO mice.
Otherwise, are you going to infect humans with COVID on purpose?
Why not? They're infecting people with COVID-19 for vaccinations and RNA vaccines from Moderna right now.
That's a purified.
I'm saying.
I know what you mean.
If we're going to talk about the ethics of it, clearly these people don't care about ethics.
Well, the patients they're not being forced to go on the clinical trials, and in fact, some of them are being compensated.
So, yeah, no, I've heard about that there were volunteers and all that, but
yeah, so
some of the stuff that the Gates folks were doing in Africa, in India, and getting in trouble for some of that stuff.
Dr. Dunbar, how are you doing? He said transgenic mice. So these mice are, yes, that's what that means. So right here, they just gave some highlights, because this paper is pretty heavy. As you can see, it's 15 pages long. And the highlights are adenovirus transduction of human ACE2 enables SARS-CoV-2 infection of BOV-C mice. BOV-C mice are immunocompromised white mice. High levels of viral RNA and infectious SARS-CoV-2 accumulated in the lungs, specifically other organs as well, about 6 other organs, but specifically in the lungs, which is what they were looking for, based on the high infection rate of the lungs in humans. The next thing they said were the mice transduced with human ACE2 developed viral pneumonia after SARS-CoV-2 infection. And lastly, neutralizing monoclonal antibodies protect from SARS-CoV-2 induced lung infection and inflammation. So that's important to note. Like, these mice were healthy before, and these mice don't have a working immune system. So it's basically a model, like you have a person who an older person, a person who has pre-existing conditions that would affect their immune system, all of that. That's the type of mouse they're using. So hold on, there's one little thing. Okay. So here's some of the figures. I'm not gonna go through all of them, but some of them are important to look at. The weight change. So look at all the different groups of mice they have to use to do the study. And you have a control for every group. Every color has a control, which is overkill for a study like this. biopsy naive, biopsy naive, or DBA 2J naive. Then you have the control and treatment, control treatment, and then you got a negative control down here. So STAT1 is a immune system regulating gene, and it's a knockout. Negative is knockout. Transgenic mice. That's what they are. They're genetically modified mice. AG129, I think that's a mutant as well, mutant strain. DBA 2J is mutant strain. BOB-C, this is regular. The black is regular. Usually that's what that is, black here as well, regular. Just regular mice. Naive means they have not seen the virus, and the other one, they've seen the virus. Mutant, they have no virus. Then the mutant has seen the virus. And so they show the weight changes, and weight changes didn't seem to be significantly different, but like for instance, DBA 2J naive, is the control, it dropped. When you give them the infection, it goes down. RAG is around the middle. The RAG negative, homozygous negative or recessive mice is right here around AG129 with infection. So these are not infected. And so it's just showing weight changes. Since that's another thing that humans go through, patients go through, is changes in weight. You drop weight after infection. That's what I've been hearing. So here they show.
So this is showing the actual causality for the effects that are being exhibited, the effects and symptoms, and the ever-growing symptom list of the symptoms that are being expressed in humans. They're able to ascertain that based on this GMO mouse study.
Well, yeah, because if you target genes that you see go up in patients in the mouse, then you can, and you see weight changes similar to what you see in patients with those same gene issues, then yeah, that's like that's reproducible. So I mean, each of these circles, each of these dots for the days after inoculation, so 1, 2, 3, 4, 5, all the way up to 7 days after they were given SARS-2, each of these dots is probably, it's got to be like at least 3 mice that they're testing for in each dot, each group.
So I just posted in the comments that from this site, the Koch postulates thing, it's like the microorganism must be found in disease, but not healthy individuals. The microorganism must be cultured from the diseased individual. Inoculation of a healthy individual with the cultured microorganism must be must recapitulate the disease. And finally, the microorganism must be re-isolated from the inoculated diseased individual and matched to the original microorganism. So yeah, so it's like the round, the sort of round robin isolation process of elimination. It just doesn't seem like that's happening even with this study.
It seems like because like BGS just said, Koch postulate, it's been superseded in this particular study and several other studies. It's been superseded because like I said, they chose the mice for a reason. These mice do not have ACE2 at all. They don't express that gene. It's not there. When you sequence the genome of these mice, the BOB-C strain, they do not have that gene at all. They don't need that gene. They're immunocompromised, and they don't have it. So you have to put that gene in there. And you know the sequence of human ACE2 because we have the sequence across the board for many patients for ACE2 receptor. So all you have to do is isolate that sequence, take that sequence, put it into a vector, a virus, the adenovirus, and that is the ACE2 gene in those mice. Now they have that gene in there. So you can track how that gene mutates after infection. You can track what happens to that gene expression. Does it go up after infection? Does it go down after infection? That's the same thing they're doing in patients right now. They're looking and seeing, does ACE2 go up and down in these patients? There was one study that said that ACE2 goes up. There was another study that said ACE2 goes down. There was a few studies that said that certain immunoglobulins actually increased after infection.
I guess based on what I've been reading about this in these 4 postulates, I guess I don't understand why is it that in this particular case, like they get to divert from this, or because this, from my understanding, the whole concept of virology and relative to germ theory, virology, the whole industry in science around that kind of centers around Koch's postulates and these exact steps, because it is a pretty thorough process of elimination. But it doesn't seem like these studies are actually doing what's here. And then BGS said they can be superseded. You're saying that too. it almost
Yeah.
sounds like somebody came up with a convenient workaround.
No,
No?
not necessarily. I mean, I have, there's another thing. Oh, actually here, this is important too. There's another, because the second part of the figure, they just reiterate what they just did earlier. This is tissue. So you remember I was telling you when you do like any kind of staining or like with patient tissues, which is what they would do, or any kind of viral, imaging, microscopy and stuff like that, it's already dead. So these tissues are from dead mice. And what this shows is you don't have any adenovirus. So this is normal. Alpha IFNAR1, this is no adenovirus, no infection of adenovirus control. And then alpha IFNAR1, adenovirus, human ACE2, all of these have infection and they all express ACE2 of human strain. And then you have day 0, day 1, and day 3 after COVID infection through the nose, because they gave them intranasal spray through the nose. And so these are the broad images, from way back from a distance. And then these are the close-up images. And so day 0, you see the infection. Anything that's brown, the darker brown is more intense staining of ACE2. Because ACE2, there's an antibody for that you can buy from the companies. And then here, you see how it's brown. And day 1, 24 hours later, it looks almost black. These dark black, brown staining. And then day 3, you still see this dark color that's there. And this is controlled, so there's nothing. There's no staining there. And then we come down to the last panel. You see, of course, there's no staining here, but you do have brown staining here in the tissues. So all this is showing is that SARS-2 infection in conventional laboratory strains of mice and expression of human ACE2 after transduction. So that tells you they were able to put ACE2 into the mice, and it was not there before, because this is a mouse that doesn't express ACE2. They stain the tissue with the antibody, but there's no activity because they don't have it. They don't express that gene. So that's important. Another thing that they did, I'm coming down some more. So there's more here, weight change, even more groups and stuff. This one, now, this is figure number, let me come down here. Where is it? I think it's figure 3, if I'm not mistaken. I think it's figure 3. So they come down and this is SARS-CoV-2 viral RNA. And this is 4 days post-infection. That's all this is. 4 days after the infection through the nose, they have lung, heart, brain, liver, spleen, kidney, duodenum, ileum, colon, and serum from the blood. And this is log base 10, number of copies per milligram or microliter. So the lung has the highest. So this is the virus in the lung. This is the virus in the heart. You have brain, you have liver, and liver, it's kind of curved over, and that's based on the different groups. So adenovirus only is black, it doesn't change. Alpha IFNAR1 plus adenovirus is another control, and then the red one is the most important. So if you follow the red dot throughout this whole thing, you can see it's high for these 5 organs. It's the highest for lung. And that's exactly what they see in patients. The first 4 are what you see in patients now, because I remember a couple months ago, they talked about people having brain fog and having issues with concentration in the brain. That's affecting their nervous system. And so this right here, this image right here, to me, it speaks volumes, but I don't know. What do you think? No.
I'm not with some of the stuff with the markers and all that. I don't know. I mean, I've just been looking at it through kind of the lens of just the stuff. And again, there's still so much. I literally just got this book, Virus Mania: How the Medical Industry Continually Invents Epidemics, Making billion-dollar profits at our expense. This other one from Trebing, Goodbye Germ Theory. And I got this other one from Don Lester, David Parker, What Really Makes You Ill. And again, they're basically just coming at the whole foundational concept of germ theory versus terrain theory. And Jennifer Daniels, Robert O. Young, Andy Kaufman, and some other names that they were firing off, just doing these different. Stefan Lanka, I think his name is, German scientist. I was looking at some other German scientists who were just basically saying that, and to BGS's point, as we've progressed in 100 years, we're realizing that we, using that funny phrase everybody says, we understand, or the understanding of this science has made some breakthroughs, but they're in opposition of the current understanding that has, a lot of people's careers and a lot of money and industry has been established on this, not to mention the control mechanism potentiality. So that it stands to reason that it's like too big to fail situation where nobody wants to rethink it and have to undo this. Plus, it starts raising other questions like, well, wait a minute. If it is true that what we're understanding as or identifying as viruses is actually something that's endogenous to our bodies, and it's part of ultimately a detox system, a cleanup system, and that maybe we rethought we need to rethink the whole thing and got it wrong a long time ago, and that we should be trying to rethink it. It sounds almost like there is almost a conspiracy of sorts to keep it a certain way. And then when we apply that to what we're experiencing now with this COVID-19 thing, if it has, I don't know, it seems kind of murky based on, GMO mice, all these things to isolate this stuff. These other doctors, microbiologists are outright saying it hasn't met Koch's postulates. I don't know. Again, I do want to show you that presentation he said, but I'm just looking at it from, yeah, I definitely want you to see it because it's super interesting stuff. But in some of these other discussions with some of these doctors and how they're breaking it down, again, I don't know this stuff. I'm 5 months in on the most virus information. I'm looking at all this stuff and it's super interesting. And again, I think it's kind of advantageous that I have that sort of blank slate where I can just keep asking questions and look at it. But then I can also say, well, look at what's actually happening to the world as a result of it. So it does stand to reason that if somebody did want to take advantage of the lack of our information or take advantage of science not being allowed to be fully expressed, or maybe there's suppressed science, that it's working to the benefit of people that stand to make billions or trillions off vaccines. Some of the what we're seeing with the government's kind of encroaching on rights, encroaching on the Constitution. The idea of the money that's being made, like it's shifting, the wealth gap is expanding. A lot of people are losing their businesses and their livelihoods, like the economic reset sort of stuff that's being discussed. This does seem like a setup for that. So I kind of look at it like, well, wait a minute.
Yeah.
If there are people proposing other ideas with science that is maybe suppressed, I think we should be looking into that. How come Bill Gates isn't looking into terrain theory? Like maybe there's something there. Maybe we don't have to use something as invasive as injecting people with vaccines that have MRC5. Like, why do we want to put fetal embryo material inside of people in big protein forms and aerosol mercury? Why do we want to put that directly in people's bloodstream? Like, that's just crazy to me. I don't know. This stuff is just weird.
That's one thing I wanted you to mention, and Dr. Dunbar mentioned it. He said that, can we get an explanation of terrain theory? I'm not familiar with it, and I'm not familiar with that either. So can you help me with that, understanding that?
Oh, man. I mean, I have this book right here, but I don't want to.
Is that dealing with viruses or just infections in general? Well,
I think what they're saying with terrain theory is more like because the idea is this. People are saying, I have a friend of mine that I've known for 15 years. He went on Facebook and said, hey guys, I was just sick for a few weeks and I tested positive for COVID-19. And I'm like, okay, that's interesting. And then he'll say, I can tell you for sure, this virus is real. And I'm like, well, that's interesting to say that because I got sick in March and it felt like an interesting sort of sickness, like flu symptoms, but just in some interesting sort of ways. Now, I could have, in theory, went to a doctor and they said, oh, it's COVID-19. You have that new thing. You need to quarantine, yada, yada, yada. But based on the way I think and my understanding of just sort of the autonomy kind of idea of, I don't trust doctors. And I do understand that they can pretty tell you whatever they want to tell you because they know you can't verify it for yourself. What I'm getting at is this. Symptoms is one thing, what's actually causing the symptoms could be a whole different story. You see what I mean? So is it possible that it's how I'm treating my body? When I know I go out, I'm drinking, I'm turning up, I'm partying, I'm eating shitty food at night. I do that for a few weeks. I'm stressed out from work. I'm driving, I'm in traffic, I'm inhaling toxin fumes, all these insults to my body. Is it possible that at a certain point I'm purging that all out? That's the terrain idea, like the damage caused to your body from multiple things, your environmental stresses, your even free radicals from your own body's digestion process. All of these things working in concert to cause certain issues with your body, and then your cells deteriorating. And could it be that we're mistaking our body's natural detoxification process for some type of viral symptomology, some type of viral
Well,
causality? And that it's
you're
just a
saying
matter
it's,
of us changing how we eat or our environment. I don't know.
so you're saying that because I mean we're doing, I mean health wise compared to where we were before, the life expectancy is much higher. People are living much longer, and you know, sanitation and healthcare is not perfect, but it's much better than it was 50, especially I mean even, especially 100 years ago, 150 years ago, even 30 years ago. It's much better now than it was then. But as far as and then you have to think of the trade-offs when you have more technology or you have more people, you have denser populations. You know, there's a trade-off with that. So I don't know.
I mean, I was just breaking that
I'll have
down,
to look
all of
it
that
up to
relative
see.
to the idea of terrain theory. Somebody said that in here. A healthy terrain will not provide breeding ground for pathogens. And that's part of the terrain theory concept, that is more or less things that you're doing to yourself, your conditions that you're placing on your body that allow for even like a bacterial overgrowth. Because obviously our body has bacteria. My understanding is there's more bacterial cells than there are actual human cells, and bacteria are a part of our biology. We need that for our gut flora. We need that for certain bacteria on our skin. Now, when you get into certain funguses, my understanding, certain funguses are actually supposed to be there, but there can be fungal overgrowths based on certain imbalance. You eat too much sugar, you can have candida overgrowth. Like those kind of
Yeah,
things start to make a lot more sense. So
but you create that environment that may be more um, helpful for the virus to grow. That's what they mean by the pre-existing conditions. You know, high blood pressure, obesity, the diet, which leads to obesity or diabetes or anything like that. They're not just, I mean, diabetes is a wide concept. It's not just type 2. It could be gestational diabetes when a woman is pregnant. It could be type 1 diabetes when a person is born with it. So there are a lot of pre-existing conditions to that. That all sets the stage for I would say hypothetically, it sets the stage for a viral infection to get worse. However, with COVID, it's a totally different thing. You got people with but no pre-existing conditions that have COVID or that are dead, who had no pre-existing conditions. And I'm gonna have to look more into the terrain theory because it seems like something that, you know, maybe needs more investigation or more questioning. Like I always want to ask
part of
questions
what it
because
is I'm at a loss to describe it because I'm still learning more about it. So this is just my mediocre version of getting it down. But somebody in the chat, I see, is saying some interesting things. Pre-existing condition equals unhealthy terrain. So that's part of the. Apparently, they've been reading up on this stuff because they're
yeah.
citing a lot of the premises of the idea of terrain theory versus germ theory. So the idea is that it's essentially one or the other, meaning we know there's certain microbes, there's certain germs, but when it comes specifically to the idea of viruses, that they simply may have been mislabeled, because we know there's bacteria in our body, we know there's funguses, we know there can be parasites, of course, and then even the parasites eating whatever in your body and them defecating in your body can cause even other chemicals and pathologies and all that. So then that could even set the stage for certain symptoms that could even be then claimed to be virus or viral load. You see what I mean?
Yeah.
Like,
I mean, I look at viruses as, oh, go ahead.
I was just saying, if this is the case and you're starting from a flawed, not you, I'm just saying, if that's the case and we're starting from a flawed premise, and keep trying to filter everything through a flawed premise, then we're going to keep arriving at potentially a flawed situation. Meaning, if we're insistent upon the idea that there are these inert viral particle things that come from outside, get in you, make you sick, if you're going to filter it through that theory, germ theory relative to viruses, and try to apply terrain theory, then you're probably going to end up at a stalemate or some type of confusion. Because I guess the idea would be try to approach people's illnesses and symptoms and remove the whole concept of viruses as we've always been taught. Now, I guess that would be a hard concept if you've been like trained in such a way, or anybody who has, or if your career depends on it. You know what I mean? To that greater point we were saying, as far as getting funding and all that, like, yeah, if somebody's livelihood and paying student loan debt and all this stuff relies on the idea of I need to stay with the accepted mainstream concept that viruses are what they are, I'm not really gonna try to go look into terrain theory or something alternative to that, because not only might it be a career ender, there's no money in it. You know what I'm saying? Like, just from a purely objective standpoint. But that would ultimately be suppression of a potential alternative mode of science. But the crazy part is, if there's something there, if there's something we can take from that, maybe there is some type of actual thing that we can use to bolster our immune system, bolster our terrain, that would negate the need for vaccines, that would eliminate viruses if they are there before they start, which would eliminate bacterial overgrowth or fungal overgrowth just by focusing on our terrain. Interesting, because the medical industry focuses a lot more on treatments and not cures. They rarely talk about preventative maintenance, what you're eating. At the same time, there's a lot of funding for FDA approvals for shady food, GMO, all this type of stuff. I mean, if we start to step back and look at it all together, we'll see holistically we're kind of being set up for failure. So it makes perfect sense that they would suppress terrain theory, because that's something you can actually affect yourself and empower yourself. And at the time, these corporations would be making billions of dollars trying to sell you some white powder chemical substance, God knows what, or inject you with thimerosal or aborted fetal tissue cells. Like, it just ends all that.
Yeah, I mean, like, BGS, you know, you can come up here, because anybody can come up here. I was asking Double C earlier about what he meant, but it passed. I can't see it. But anyway, yeah, like I said, I have to look up and see the terrain theory, because my thing is, if you're talking about terrain theory and that is the culmination of everything that negatively will affect you or positively affect you that's going on in your environment, then you can't get mad when technology companies develop meat that may or may not be what you're used to, because there's a shortage or there's a potential shortage in the future. You know, like I said earlier, you have trade-offs. So if growing food hydroponically or growing food any other kind of way that's more efficient with more nutrients is the best way to go at that time, and there's technology involved, but if you acting like a Luddite and you don't want any new technology, well, then you're gonna have to starve, because that's the change in your terrain that may be to your benefit if you want to survive. And so I look at it like that, because even though it may be a virus, you know, it may be GMO, it may be altered, and you know, that would prevent you from getting infected by a certain virus, but you have to think about that.
Yeah, and that's the thing. Like, I don't want to misquote the whole idea, because there's certain tenets to the whole premise of terrain theory, but I think one of the main things is relative, more or less, to what you put in your body. So the idea of eating like whole foods and not processed foods and things that would create something like bacterial overgrowth. Because, you know, a lot of people are focusing on that more, like your disease starts in the gut. Your gut health is going to dictate a lot of other things. The idea of, you know, the connection between brain cells and similar brain cell neuron type cells, even in your gut. So there's that whole, you know, I had a gut feeling about it, that whole concept. So there's that, you know, interconnection, holistic nature of your body as this, you know, interconnected system. And that if you ingest the wrong things that short circuit, you know, this particular organ, then you leave yourself exposed to certain bacterial overgrowths or fungal overgrowths that set you up for failure, or you work at a chemical plant and you touch a certain chemical or it spills on you and then it makes you sick and then you start throwing up or you die or your lung collapses or something crazy like that. But then, you know what I mean? Like, so I don't want to muddy it up, but I'll try to find everything I can to have you look at some of what I was looking at with this, the terrain theory concept, and even more of these presentations on could viruses have been mistaken for exosomes? So that's an interesting thing, which is a similar but kind of separate theory in and of itself, a different idea, I think. Again,
what I showed you earlier with the exosomes, I was going to say that with Dr. Hildreth and Dr. Gould and a few other doctors, these are physicians, and they're very well respected even to this day. Dr. Fauci as well, you know, many other professors and doctors, and especially during that time with HIV. So they were saying with the exosomes that the virus is not an exosome, that it uses exosomes for transmission.
Yeah, as a transmission vector because it is traveling.
If you can now the electron microscope can actually see down to 1 nanometers, right? Okay.
Yeah.
You can see the thing. In other words, you put them side by side on a slide. You can see what's the virus, what's the exosome. It's not that you have to guess. It's not a guess. You can see it.
Yeah, and you know it's inside of an exosome. So at the time they didn't know, they didn't have the technology, but he said that this is acting like an exosome because they didn't even understand what exosomes really were at the time. And so they were trying to figure that out. That's what that article, I just put it in the chat earlier. That's what the article was trying to figure out. Dr. Hildreth may have been wrong, but he never said it is. Like, he never had publications that say, I looked it up. He never had publications that say this is what HIV is. He was like, this is what it seems to me. This is a hypothesis. They hypothesized. Sure. Then they found out like exosomes are used for traffic, you know, transferring information from place to place. And that's what the virus is using as a cover, like using it as a taxi in a way.
What I'm saying is they make circuits in your computers that you can buy right now that are smaller than viruses. They can see the thing. Okay, it's not like you have to guess. You can isolate it by seeing it. With COVID, the first thing they do is took the particles and basically grew it. They actually regrew the doggone thing from scratch to prove it. This is not something, like I said, with COVID. This is not something that's new. They've had documents going way back to 2006 with this particular virus. The Chinese destroyed 15,000 cell lines. Of this thing. It's not something that they mistake. They poured a lot of money into this. Okay, a lot of money into this. This is not something. This is not something. That's not something on the outside. How much money can you make on a vaccine worldwide? $1 trillion. You spent that. You spent that in the month of March alone. This is not a thing to make money. This is actually breaking up your system. Okay, this is a royal screw up. This could bring down your whole entire system. In other words, I don't care who you are, how you lead, okay, what you're in charge of. If the whole system breaks down and turns into chaos, you lead nothing.
Yeah.
Sure.
Yeah. See, I wasn't going to, argue against that point because I know that you're still, I know that you're still learning and you're trying to figure this out. I was hoping that showing the data, like showing the figures, like this is showing you this mice does not have this gene at all. It does not make the gene.
So as a kid, because I'm a trained chemist, right? Okay. What you do is you need a control, right? So you take something that didn't have it with its receptor, inject the doggone thing into it and see what happens. Right. If nothing happens, okay, now we can actually go out and re-engineer that mice with your requirements into that mice and do the same thing. If it happens, we know what we introduced into that mouse. It's a controlled experiment.
Yes,
that's actually an interesting thing. It does seem like it works as a control. I mean, I don't know. Again, I would have to look more into that.
You do the same thing in physics. You do the same thing in chemistry. You do the same thing in mathematics. Basically, you introduce a controlled experiment. That's exactly what they did. In other words, you find something that didn't have it. So in other words, if it didn't have it, in other words, you want to see if the H2 receptor is what the virus binds to, right? So the first thing you try to do is find something without it. In other words, a sterile environment. Inject it into it. Okay, if it doesn't exhibit any kind of disease and the virus doesn't grow or bind or anything like that, then you know that the possibility that the ACE2 receptor is what it binds to. So then what you do? Okay, you want to find out the opposite. Okay, so now you introduce something with an ACE2 receptor into it, right?
Sure,
The same thing. The same organism.
You just explained that exact same thing, and I get where you're going with that, but I'm still trying to wrap my mind around the whole idea of how it was actually isolated and shown to be a living organism that is the cause of the particular symptoms that are being exhibited as that disease in the first place, and being able to be cultured outside of that organism.
Let me.
I'm just going by Koch postulates.
Okay, hold on a second. Okay, just like anything, right? If you take a living organism without it, right? You take a snapshot of it without it, right? You look at everything that's in that slide, right? Without it, right? And then something gets introduced into it. Can you not tell the difference in the picture?
You can see. I'm looking at the picture right now.
You can see it.
I mean,
it doesn't have to be living, right? Okay, take something like a fossil, right? Okay, you take bare ground that doesn't have elephant bones or whatever it is in it, right? Or certain kind of rocks in it.
Right.
Okay.
Wait. So let me hold on. Let me wrap my mind around this. So snapshot, healthy organism. Next snapshot, there's this new particle there, and they're exhibiting some type of symptoms. So by that evidence, we can dictate that particular particle is the causality of those symptoms.
That's one part. In other words, okay, if you take a healthy organism, right? It doesn't have any of those symptoms, and then you take another organism, the same organism that does exhibit the symptoms. What changed?
They have symptoms.
They have to change. Something changed. You have to find out what changed.
Yeah.
That's a scientific method.
I'm following that. I'm following that. So then I'm just trying to, again, I don't know this. You guys know this stuff way better than me. So I'm just trying to run this through limited brain and understanding of this. So healthy, there's some type of symptoms. We get another snapshot. There's something there. So we start to hypothesize that this particular thing is the causality of those symptoms.
Yeah, because that's the only thing that's been introduced.
Right. Okay. So then the idea from there would be to take that exact thing that we are assuming is the cause of those symptoms, and then put it in another healthy organism and see if it exhibits those same symptoms.
And see if it does the same thing. That's what it's called. Yeah. That's called repeatability. That's the same.
Then here's the part though, as far as, because again,
yes.
So then there's that middle step as far as with Koch's postulates, being able to culture it even outside of that living culture of it. That's the part.
If you look at the screen, you'll see it is not sketchy. It is not sketchy. It's on the screen.
Or I just mean
no. This is histopathology, and Dr. Dunbar knows what this is. This is lung sections. It's from the paper. I put the link in the chat. This is histopathology. Histopathology shows you the stained tissue from an organ, a slice, like 5 micron slice from a tissue from the lung of the mice, and they stained it with hematoxylin and eosin. So one is for nuclear. Anything you see like little dots and stuff, nuclear staining, little dots. So this is lung tissue. This is no treatment, 0 days post-infection. And then you have so there's no treatment there. There's nothing, no virus, nothing. Adenovirus is a baseline control virus. That's how the lung tissue looks. And it's showing you the broad spectrum image here, and then it's going into more magnification. Then it comes down 4 days post-infection. And this is actual adenovirus and SARS-2, COVID-2 right here. Look how the tissue's looking. It looks bad compared to control. I mean, it's looking less differentiated. When you have less differentiation in your lungs, you have inflammation. You're going to die. The reason why you don't have like longer than 8 days, this is 8 days post-infection. Look at this. Look how the lungs look. It's all messed up and torn up. You don't have the connection. You have holes. These are holes in the lung tissue. Look how the lungs looking. There are literally these big gaps. Those are holes in your lungs where the virus is just tearing the lungs up, basically. And I mean, this is the worst.
I have to like watch this.
You know, there's a reason.
I'm still trying to make the connection to where it's known
8 days
unanimously
post-infection
for
is the
sure.
highest they have. You know why 8 days post-infection is the highest they have? Because after 8 days, they're dead. They don't have any
It
months
just
after
seems
8
like
days.
some of this is almost like assumed or almost circumstantial. I'm not seeing
That's not
how one is
assumed.
the
That is,
actual. I'm just saying,
You look
from my
at
point of view,
circumstantial
I
is
just
when you don't
know.
have visual evidence. This is visual evidence. It's not circumstantial. It's hard
then
evidence.
and then they are very. The study was good because they have a section here that we don't even like to put. I don't even like to put in my studies limitations of the study. But since they're doing COVID, they have to put a section like this now because so many people out here with this quack medicine and mess they're doing, research they're doing. So they have a whole section. I want to let you see how long the limitation. That was a long section. Let me go back up. Roll that back.
Yeah.
Limitations of the study. That is long.
Yeah.
For them to talk about what they couldn't do.
Yeah. When I want to talk about terrain theory, the thing is that if you're going to introduce any kind of theory, it has to have actually go through the same steps and has to be proven that the same thing that happens with germ theory, right? In other words, terrain theory, what I was asking is, okay, with terrain theory, your body is a closed loop system, okay? How do you transmit your system or your exosomes that's particular to you to somebody else, or your symptoms to somebody else, and with somewhere halfway across the world with a different
I mean,
environment?
I could take a shot at trying to describe what the doctors I was looking into were saying.
Mm-hmm.
I mean, what they were saying is essentially just to take a concept that I get sick with certain symptoms, and then my little brother gets sick with certain symptoms. So I guess the idea is that it could be assumed or thought that I got sick in some sort of way, and then I coughed, and then he caught what I had, or I caught what he had, or it was transmission back and forth.
Mm-hmm.
I guess within the germ theory model, it could be thought that because we live together, we could be potentially exposed to the same mold or something in what we're eating, or something that created some type of bacterial overgrowth that related to those symptoms.
That's true. But how do you explain when somebody that's not in the same environment that you are, that's halfway around the world, gets the same thing?
That they could be exposed to a similar chemical, because we're talking about symptoms.
Yeah, well, a similar chemical. But the thing is, scientists would isolate for that.
Yeah.
That's the first thing. The first thing before you talk about it. Okay, somebody that's in some place that has mold or toxins or someplace like that, right? You can understand that, even in a city, maybe even a state or country, right? Well, if somebody in the desert that is not exposed to nothing, a completely different environment catches the same thing, how do you explain it?
Well, I guess the idea is that you're speaking from the premise of starting with the foundational concept that they are catching something. So I'm taking it back to the idea that we're saying that they're exhibiting similar symptoms. So I think that's the part that.
So in other words, very specific symptoms. Because
I don't know. That's interesting, because even with this whole COVID thing, there was this symptom that seems very similar to the flu, and then there's more symptoms that add on, then more symptoms, then more symptoms, then more symptoms.
Thing is, okay,
so it's like a catch-all kind of things where you get to say, well, yeah, pretty much anything is COVID-19 now. How convenient.
The flu has how many? Flu has how many symptoms that is associated with it just by itself?
I don't even know.
Like 20, 30 symptoms you get from flu?
At least a dozen I know of, yeah.
At least a dozen, right? Okay. And the flu has been around for 200 years, right? That we know of, right? Okay.
I don't know that. I get that somebody is saying that. That's the thing. Like, again, I'm rethinking all of this stuff.
In other words, that we know of is that they've isolated. It's been around for at least a couple hundred years.
They isolated from people who were Inuit people living in Alaska, from their bodies buried in the ground.
That's from the
that was I forget his name. Yes.
So then to know that and say that, wouldn't we have to then go look at various peer-reviewed studies that show that they isolated the flu virus, right? They got to go back and run back that same thing.
PhD dissertation.
They did.
That was his dissertation.
In fact, they recombinated the H1N1 flu in 2009. No, in 1999. Excuse me.
In 99, 2000, 2001, they were doing, because this was leading up to the Human Genome Project, and they were doing a lot of stuff with Spanish flu. Recombining it making it less virulent, and they're working on that. So if they're doing it then they definitely did this with the same thing they're doing now with SARS-2, what I was showing you earlier. They've done similar work with the first SARS strain, SARS-1. Only thing is they didn't have the technology they have now.
Yeah, with the thing is that with the terrain theory, they have, the thing is, you haven't proven transmission, but basically with germ theory, it's very easy to actually prove transmission. But the thing is, terrain theory it doesn't have any competing model, because
well, you know what, that is interesting.
In other words, you can't prove. I'm a chemist, right? You cannot prove that somebody in completely, totally different environments come up with the exact same thing, exhibiting the exact same symptoms, and actually
well,
the exact same stuff.
I think that's an interesting thing, because if you, just the way I'm thinking it, again, I'm not a trained chemist. I have nowhere near the training of you or Nicole. So I come at it from just, I don't know, maybe like this right brain sort of artist concept, to where I can abstract it and say, well, I see what you're saying. You can't prove the transmission, but I'm already starting from the standpoint that I'm not even necessarily taking it or leaving it that somebody is actually transmitting something to somebody. I can entertain the idea that maybe somebody could be exposed to mold here in Oakland, California, and somebody could be exposed to something over in China and exhibit the same symptoms. So here's the trick. When there's a certain sickness or disease or series of ailments, and then they get to say it's 30 symptoms, well, that's interesting. You might exhibit any one or more of these 30 symptoms. It stands to reason that that's pretty ambiguous, right?
Except for one thing. You can actually, with an electron microscope, you actually isolate the particle.
Now that's interesting, because I'm curious to see. I'd be curious to hear you guys. I'll try to send you guys some links. I'll be curious to see what you guys would say about these doctors and microbiologists that are saying no, Koch postulates or these things haven't been isolated. Again, I don't know. I hear what you guys are saying, but I'm hearing what some other people are saying. So
in other words, as science advanced, because Koch's postulate was made as a postulate, as a process for his time, right? With the tools that he has. Okay.
Yeah.
In other words, if we're back like even 70, 50 years ago, we'd have to use Koch's postulate because we don't have anything better, right? What happens when the electron microscope comes in? You can actually directly see these things. Recombinate them, extract them with the tools that you have. What if Koch had those same tools back 100 years ago? Was his postulate be the same?
Yes, I mean, okay.
I think they would, because it sounds like a process of elimination.
That's the issue. BGS, myself.
BGS, I almost feel like you described something that it was akin to Koch's postulates when you first popped on the stream.
Well, I agree with Koch's postulate. I think that this study and many other studies that are working on right now, that's similar to this study, where they are looking at mutating different parts of ACE2 receptor in order to make it a less likely candidate for the virus to infect. That is Koch's postulate. That's the postulate right there. This paper went through Koch's postulate backwards and forward. That's what I said before I even showed the study. That this study, it answered number 1, number 4 at the same time, and then answered number 2 and number 3. You can't. I mean,
that just actually dawned on me too.
They have the actual viral RNA copy numbers 4 days after the mice were infected with the virus. They give you all these different organs that have viral copies in the organs.
I want to see something though, because since you sent me that, and I looked at it myself, and again, like I'm 5 months into my virology studies. So I'm curious though, because there's this doctor who talks about that, and he's the one. I'll try to get it to both you guys, where he's talking about the other exosome thing. But it's curious though, because he's saying that he said he looked at a few different studies, one from Korea and some other different places, and he was like, it doesn't appear that they've actually isolated the virus, yada yada yada. I'd be curious to know if he's seen the one that you sent me, Nicole. I'm gonna actually see if I can email it to him.
Not only did they isolate, they had isolated the virus before it even escaped. It was in the lab. It was in the lab. They were growing it. They were growing it in the lab in China.
It's already sequenced. They already knew the sequence. It was an RNA genome. They know the RNA genome. They know that sequence. They know specific target sequences within the genome. They know if it were DNA, if it ever reverse transcribed to become DNA, they know that double strand sequence. They know the protein sequence from different parts of the RNA genome. So that's how they were able to do this paper, because you can, through bioinformatics, which Koch did not have at the time, and through they used to call it computational biology. Now it's called bioinformatics and biostatistics. Now you can predict what the protein expression is going to be, and it'll give you all these different, like at the top it'll say 100%, this is what the protein sequence is, and then it gives you all the way down to 1% chance the protein expression is going to be, the sequence is going to be this. And you always go with 95 to 100%. Those sequences, that's what your COVID-2 sequence is going to be, and you use that.
Yeah, basically,
you already knew. Back in February, they knew the whole everything about it.
Yeah, because the Chinese released the papers of their experiments. 2017, COVID-2, it hadn't jumped to human beings yet, but they had done papers on it. The bat lady did a paper on COVID, SARS-2 in bats back in 2017. There's paperwork going all the way back to 2006 with this stuff. They've been isolated the thing. They know what it is. It's not a mystery. There's paperwork going. I mean, there's a paper trail going all the way back to 2006.
Yeah, no, that's interesting, BJS. Like I see what you're saying with that, and again, I get to sort of have the advantage or disadvantage, depending on how you look at it, at coming at it from a blank slate, in the sense that I'm like, okay, there's information being presented by some people that I don't know, and don't know if I have any necessarily a reason I should trust any of this info. And to a further degree, the stuff that you guys are breaking down to me, and a lot of the stuff that Nicole informed me on relative to this study.
So why do you trust the books that you're reading?
I don't trust any of it. Let me tell you what I remotely trust at all is this. Me saying, okay, well, I know that when I get better rest, my body feels good and I pretty much never get sick. I know that when I eat certain types of foods, my body feels good. I feel energized. I think more clearly, my creativity flourishes, my whole mood, and then I end up getting better rest. But I know when I want to fuck off and go party and kick it and be wilding out and drinking, I end up getting sick. When I go to the doctor and they prescribe me with some kind of whatever, because of whatever ailment, usually whatever they prescribe to me makes me feel worse. So it's like my own self-science. You see what I mean? Like I've had more success with that than just going by what people are telling me.
Well, okay, you could be perfectly healthy and all that stuff and contract Ebola and it wouldn't help.
Sure. I mean, but again, I guess where I'm at is I'm just not really comfortable anymore with taking other people's word for it, because I don't trust the medical industry. I don't trust the pharmaceutical industry. I don't trust mainstream media. I don't trust government. So, I mean, to whatever advantage or disadvantage that puts me at, that's kind of what I'm comfortable with. And just sort of moving with that certain level of self-autonomy, and, you know, like, hey, it's on me. There ain't nobody out here that's showing love and got my back. Like, why should I trust these people who have tried to fuck us over on so many different levels? Like, let me bear the burden of this as much as I can, while educating myself as much as I can.
You trust them every day. You trust them to protect your goods and services. You trust them to actually provide you electricity. You trust them to actually go when you get on an airplane. You trust them when you go through security to make sure there's terrorists not on your plane. You trust them with keeping criminals away. You trust them with keeping whatever food supply, because unless you actually grow your own food in the middle of nowhere, you have to trust your food supply. Okay, you trust them. You trust them 100 times a day.
I'm referring specifically to the things that I know I have direct control over. So I think my overall message for myself is what the things I have direct control over, what little bit of freedom or autonomy I do have, I'm trying to use that to the best of my ability to empower myself, empower my body, and put myself in a position where I can be of resource and value to other people.
Yeah.
What I'm not going to do is just let somebody on some news briefing tell me this, that, and the third, and not be able to prove it for myself, and then go tout that information as if I know it's true, and add to fear and panic and destruction of people's lives around me. That's what I'm not going to do.
The thing is, what I don't understand is when something changes, right? When something changes, when a country that has nothing, that's not like ours, will pull out AK-47s and trap their populace inside, that bothers me. Okay. When they knew about this in October and they lied to us and said it was no problem up until until 6 months later when it becomes a problem, that bothers me. Okay. In other words, when I talk to people that are actually in ICUs, that do the work, that tell me way ahead of time before it even gets in the press that this is coming, okay, those are the people that I trust. Because like Nicole, when this first happened, first person I emailed was Nicole. Look into this for me. See what you say. Nicole emailed me back. Okay, this is a real thing. Okay.
Have you guys heard of BGS? You were the interesting point. They knew it was coming before it happened. Have you guys heard of Agenda 21?
Agenda 21?
Or not Agenda 21. My bad. No, not that. That's a thing. But no, it's Event 201.
Event 201.
Yeah.
Event 201. I've heard of it, and guess what? It was right after they found out the initial release from Wuhan. 2 weeks later.
My understanding is that it was right before it.
It was after it. Event 21 was in October 18th, 2019. The initial release that they found out that they guessed was in October 6th, 2019. In fact,
a few days later, they did this thing, John Hopkins with
exactly
Bill Gates, and they run this scenario that seems to mirror exactly the rollout of the COVID thing.
Because they already knew what was coming. In fact, they're saying that the Chinese are actually lying. It probably was actually released somewhere back in August, and we didn't find out about it until actually confirm it until October. In event 201 was actually after the release.
Yeah, no, that's all. That's
Event 201 is something that I've heard of, but I think I actually heard someone ask you about it on one of your streams, BGS. I don't know, because I focus on looking at experimental data and publications. I don't know what that's about, because people say, oh, Bill Gates, and I'm like, okay, he's rich, like whatever, like what is that? How is that gonna help anything? Like, oh, he's rich, he has money. There are a lot of other people who have money as well. And at the same time, like I said earlier with you, AM1, like I was saying, we can't do what we do without money. Like that study I just showed you, it costs a lot of money. That's probably somebody's grant and a couple other grants mixed in to do that research. And I still at this point don't know what Event 201 was or is. I have to look it up, I guess.
I suggest you do.
And basically, to me, it's not real till you see the bodies. When you start seeing the bodies, okay, this is real.
Yeah, for me, it was real because of the sequence and the well, I didn't see any bodies yet. I just saw the sequence, and then I started to talk to bioinformatics guys and ask them, can you cross-reference this? Can you see what this is? And they were like, it took them a long time to get back to me, and I was like, oh, it must be something bad, because if it's something quick and easy, they'll send it back to me within a few hours. But it took a day, and I was like, what the hell? What's going on? And they were like, yo, this is not good. They looked through all the different scenarios with this sequence, and they were like, this isn't, this is really bad. Is this a virus or something? And I was like, yeah. So that's when I knew, from that, I had to use, depend on somebody else to use technology, and then give me the report, that tells me, yeah, this is some bad stuff coming. I didn't see one body drop. Like, I just heard whispers about something going on in China, but it wasn't like, I got scared and started making, moves. But I was thinking still, like, it's not that big a deal. Okay, it's just a regular flu, like regular thing. Then it hit me, and I was like, wait, this is not no joke. Like I said before, all the meetings and stuff we kept having was very weird. Because some people hadn't come back from break yet.
Yeah. And then the confirmation is when the Chinese started scrubbing information from the internet.
Right, and I couldn't find certain things anymore.
Yeah,
and
before, I think it was the Indian The Indian virologist who actually said there's elements of HIV injected into this virus, and then 2 months later you can't find it. It's gone.
Hey, you guys. Hey, my sorry to cut you off, BJS. I actually have to go because I got to put out a fire for one of my client projects.
Okay.
But I want to look at this more. I'll send Nicole. I'll send you some links if you find them interesting. Just send them through the BGS. But yeah, super interesting stuff. Like I'm still again trying to wrap my mind around all of this stuff and just just looking at some of these different books. I got a whole new book collection now. And checking out some of these different doctors and kind of their presentations and whatnot, just real interesting stuff. But I just try to look at it more along the lines of how do I take this information and to the best of my ability, empower myself and not be like living in fear and like the sky is falling and all that.
The thing with any theory, you always have to run through the logic and say, where does it fall flat? And the thing is with the terrain theory, it falls flat with spread. How do you explain a broad spread in different places under different conditions? And basically you have to explain that, and they haven't.
Yeah, I don't know. I mean, there's people who are explaining this stuff way better than me. So I would say to anybody who listens to this shouldn't go by what I'm saying.
It falls flat with spread, but it also falls flat with reinfection of the same patient. It falls flat with a person who is a baby who is born with COVID, which there were 85 babies born somewhere, I think it was in Texas, born with COVID-19 infection. So it falls flat with that, because then you're going to say a woman's womb, her uterus is terrain theory. Like that makes no sense. So a lot of the things that they have with terrain theory, and I'm just going off of what I'm hearing and what I'm just glancing through right now, but a lot of it does not match the principles of a lot of basic science and concrete evidence that you have to show over and over. There are people who do research, who are doing research right now in the lab when they shouldn't be there, just to disprove a person who did a COVID-19 study in April.
Yeah.
They're doing it to disprove them because they're like, wait, that's wrong. You can't do that, And so I get it. But the terrain theory, that has something that you have to think about. If it's a theory that has held up, it would be introduced over and over and over by people who put out publications in science over and over. And I can't find anyone with this terrain theory who has a publication in science, whether it's, it may be social sciences, but in actual basic life sciences. I don't see that. That sounds like to me, honestly, it sounds like some money stuff. Like, they're gonna tell you about terrain theory, and then they're gonna ask you to put some money in collection plate, that type of thing.
No, that's the thing.
I'm not sure.
It easily makes more sense to be the other way around. That in theory, if that is true, they would suppress it because they know they can't monetize it. But I don't know. Again, please don't take what I'm saying because I'm in no way an expert about it. This is just something I'm looking into because I'm just trying to leave no stone unturned, because ultimately I can't personally verify any of this information that we've been fed. And I'm trying to do my best to understand it as much as I can, because I don't like being in a position where I have to just take somebody else's word for it and trust experts, because I don't trust any of these people. And I don't see why there's any reason any of us should.
We do
well,
all,
if you don't believe what I
time.
just presented, if you don't believe what I just presented to you, that is their data. Like, if they have published this data, they're probably going to present it in a meeting or, whatever. I know they're doing some more stuff with mutation. Like I said, they're going to be mutating because I spoke on this one time with Rob and Jade, where they want to do prophylactic studies where they can prevent infection and do that in mice. And they want to mutate the ACE2 receptor in order to prevent infection, and then look at the effects of the virus and work backwards. That's also Koch's postulates, where you can do something forward and reverse.
Some of it is going over my head because I've never heard the phrase or the connotation H2. So what I mean by believing it is I want to go back and try to do more research myself. So that's just
the number one gene out there.
Not just take his word for it.
No, you don't have to take my word for it. Just you look up ACE2. It's ACE2. Look up that receptor gene and type in February 2020. And it will come up.
Okay, no, I want to look more into all of that, because that's just a whole different level of what I've even been looking into. So now that you said that to me, I'm about to go on a feeding frenzy trying to sort that all out too. So it's just kind of my like curiosity gets the best of me, and I want to look into all this stuff because I'm just saying I don't like being in the position to where I have to take somebody else's word for it, because I think that's a dangerous position to be in. So now it makes me say, well, I want to go learn all about what Nicole told me about. So then if I can look at that myself and arrive at the same conclusion you did, then great. But I want to be able to do it the same way you did it, not just hear you say it or BGS say it and say, oh, well, now I believe it and will tout it because somebody told me.
That's why I bring the links. That's why I share.
You take just like it's competition theory. You take competing interests and see what they say and see what's in the middle.
Right. No, exactly. No, I feel that.
China doesn't care about selling. China doesn't care about selling vaccines. They don't care about that. Okay. No. So South Korea doesn't care about selling vaccines. Japan doesn't care about selling vaccines. They don't care. Okay. So why are they more strongly reacting to it than we are?
I don't know. I mean, you're asking me a question, but the premise of your question, I can't even verify because that's just something you're saying. I don't know if that's true, that they're not trying to sell that.
That's why I asked the question. I didn't give you the answer. I asked you a question so you can actually go look for it.
Oh, okay. Yeah, no, I see. Yeah, no. So that why wouldn't they be trying to sell it if they can profit from it? That's a good question.
And why is 87,000 Brazilians dead over terrain theory?
Yeah. With the same thing that they can identify, why are all these people in India and Brazil? They've already compared the genetics, the copies, and the sequences of the virus. And I already had a video that showed in India and Brazil, and especially in India, that you have multiple mutations within patients in India. And the base strain or the original copy strain of the virus is identical to Brazil. That does not explain terrain theory unless we're still in Pangea or something.
Yeah, no, I don't know.
To me, like how it can go across the water and all that. That doesn't make sense because you have people that have in India. I mean, they know they have the mutation. And they don't know the original strain.
I don't want to feel like I'm messing it up for you, Nicole, because please don't take my limited breakdown of terrain theory and start making any preliminary assertions about how it works, because I'm not breaking it down to the best ability.
You're not the only one. That's like I was actually interested in this video because I have several friends that our friendship is kind of waning because they're sticking to this stuff without proof.
Well, I would say that basically everybody is sticking to the whole narrative without proof. So everybody's in this boat.
What I'm saying is, okay, they show you images. Okay, that's proof. That's evidence, right? They show you bodies of them burying people all over the world. That's true. Unless you want to, you can actually
bodies that died does not tell you what killed those people.
You can test the tissues. They're doing studies right now in hospitals around the country, around the world, actually. I just heard about it. But they're doing studies all over the place where they're testing the employees that work at hospitals and different like research facilities because they have COVID, and you can tell from the blood. That's why they take the blood. It's because they know you might die. And
unless you're in the ICU, unless you have the expertise to actually test it yourself, you have to take people's word for it at some point.
Yeah, and I think that puts everybody at an extreme disadvantage, and that's all I'm coming from. So to the best of my ability, I want to try to sort it out, which is exactly why I would bother to even have this discussion.
We take people's word and expertise for granted every day.
Yeah, but in this particular case, so are you advocating against the idea of me trying to inform myself as best as I can?
No. What I'm saying is,
definitely look for
what I'm going to do.
What I'm saying is, you're saying that you can't. You're at a disadvantage when you take people's word for it. But the thing is, this whole system is so complex. You take people's word for it every day, and you don't even know it.
Now, last November, 2019 November. Actually, for me, it was October, around the 13th, 14th of October. We are mandatory. We have to get flu vaccine. Nobody questioned shit. Excuse me. Nobody questioned it. Y'all know how I get. I ain't gonna cuss too much.
Nobody
questioned it when it was going on. We had standard flu, which is H1N1, which is Spanish flu. Okay, nobody questioned it. When people was dying left and right, 2018, it was like 2,000 people every week was dying, dropping dead. Nobody questioned that. Nobody was questioning when we got the flu vaccine in 2018, and this shit didn't work, and I still got sick and had to go to the hospital. Nobody questioned this stuff. I didn't even question it. I was like, you know what? I gotta do better. And I mean, I was sick as a dog, 2018. I remember that. 2019, last year, right before COVID, nobody was questioning the flu vaccine. Nobody was questioning the flu. And is this a real thing? Nobody, if people didn't even know that's the flu from 1918, they didn't even know that at the time. So like all these other things that's going on, Ebola, nobody questioned that. The first SARS, nobody questioned that. Why all of a sudden people questioning SARS too? Because they had a meeting somewhere with some white folks with money.
It's probably. I'm thinking maybe they're questioning it more because we're seeing a whole economic collapse and a lot of people out of work, and
that should make you even more likely to adhere to what's going on from the medical community, because you're gonna be sick because if people ain't got no money, so your likelihood of getting sick is much higher if you don't have money to get the right food that you need. In my opinion, like when my grandmother, she wasn't alive at the time, but her grandmother said during the Spanish flu or epidemic that they had, she said people were dying left and right, and the last thing people did was question. I asked her, 93 years old, I asked her back in March, did people question the government back then? Do you know? She said her mom was like, no, they had to go stand behind them trucks and get the food. That didn't happen. They pretty much swooped in and was like, hey, we're gonna help y'all, give y'all a little money, so y'all can keep it moving. But in the midst of all that, if you're gonna question anything, question that, not the fact that people dropping dead and you can see the bodies and you can, I'm literally showing the pictures of the virus, like of the infection in the tissue of the mice that
that's the thing. I think
have virus.
Relative to what you're showing me, all I'm saying is I just want to take, like, I'm gonna rewatch this video and then try to sort out the ACE2. I want to go learn all of this to where it's not just the point of you showing me it within a couple of hours. I don't think it's a smart move for me to say I have confirmation of it just because I saw you do a presentation in a couple of hours. I don't think that's a logical way. Like, just observations, I have now proven it. I don't think that's logical for anybody to expect such a thing. I think it is logical for people to question things, especially something coming from a known corrupt government, known corrupt medical organizations. Iatrogenic deaths. There's more iatrogenic deaths as reported by
all 200 governments.
Yeah, you can't publish that type of stuff without proof.
But not America is corrupt. Okay, Canada corrupt, Mexico corrupt, but all 200 governments, all of them.
I don't see any reason why not.
They're all corrupt.
Hey, you guys, I gotta go though. I appreciate it. I want to do this at some point again because I want to try to get some more info and kind of compare notes more.
Rodney works in the ICU in Arkansas. He's been telling me from the beginning about the symptoms because he works with these people.
Yeah.
Hey, Rodney. Yeah, I wish you could have came on earlier. But yeah, I got to go handle this stuff. All right. But I want to chat with you guys again.
Thank you, everyone. Be safe.
All right, y'all.
All right.
That's what we're up against.
Yeah, this is amazing. I caught like the tail end, but I mean, I'm telling you, look, I just got another one. I've got 3 more over the last few days, and I can tell you, the symptoms vary. I mean, the last one that just went out, asymptomatic, except for respiratory symptoms. But they pretty much manifested over the last, I don't know, couple of days. That's not me.
Okay, I thought you would give it so she wouldn't come down.
I might.
Oh, okay.
I might. Sorry, y'all.
It's okay.
But yeah, but it's hard to convince these people when they have an answer or to every position that you put forward. So if you're doing that and they say, well, what if it was space aliens, and you can't really come back with anything else. I mean, look, even if you showed it to them under a microscope in tissue samples, they still wouldn't believe you because they don't know what the hell they're looking at. You'd have to take them through.
I'm telling them.
Yeah,
I'm
you'd have
telling
to.
them.
But they don't believe you because
They don't believe you
verify
because they'd have to be able to do it themselves. They'd have to, but then you'd have to take them through cell biology, micro, virology. You'd have to take her through all, parasitology. You have to take them through everything just so they can understand the intricacies, the small things that will make them be able to connect the dots. So, and these people, unfortunately, we live in an environment now where they have way too much information and not enough ability to be able to interpret that information.
Yeah, that's the problem.
Yeah. And that's the problem. I mean, imagine if you were in a college and you're telling you are trying to debate your professor with that type of logic, with that type of argument.
Yeah. The thing is that it's not just towards him. The thing I don't like is when they throw a competing theory at you, right? So you ask me, okay, tell me about the theory. Well, I'm not completely versed in the theory, so why are you using something that you don't
know about?
Yeah, you don't know about.
Absolutely.
And my thing is, why would you, when in the comment section, I knew what Koch's postulate was, and I knew of the study when I saw it and read it in the comments. I said, "Oh, I know the study that goes with that," and I just put it. That's why I posted it like so fast. And then he asked about exosomes. I posted that too. But see, the exosome study or the exosome question has not been proven for HIV. It has not been proven for that. Exosomes have been shown for cancer. They've been shown for other things, but not HIV specifically. Dr. Hildreth is a very intelligent man. He knows what he's talking about. At the time, that was a hypothesis. Koch's postulate is for that time. But what gets me is it still fits in 2020.
Yeah, for certain things.
That's it. Some things work and some things don't. That's what science is about. It's intense study, figuring out what's the truth. And the truth is, I was saying Koch's postulate is true, and he's saying that it was not answering Koch's postulate. But I'm showing you and like evidence of where it did. I was just like, I don't know what else to say because
because he didn't really understand. He really doesn't understand Koch's postulate. He's just using it as an argument.
Oh, I'm gonna mute you, Rodney, real quick.
Okay, I'm sorry. I'll mute you.
No, that's okay.
That's because I have a friend that's using the exact same thing. I said, okay, say that exosomes are true. Say that the terrain theory is true. It said, how do you transmit your symptoms from one person to the next, or around the world that's in a different environment? Okay. Okay, how do you investigate and say why these two people in separate environments got the same thing? Okay. The thing is, you have to be able to prove that. You can't say, well, maybe. No, it can't be a maybe. You have to prove it. You have to show causality. If you're gonna debunk a theory, you have to show causality of a superior theory. It doesn't work that way in science.
Yeah, it just seemed like skepticism to a fault at that point, where it's just like you basically laid out as much information as far as evidence is concerned, but he just needed to see it for himself, and even to see it for himself as far as bodies is concerned, and even he needed more evidence than that. It's like, okay,
I don't know what they died from. They just
exactly.
You're gonna go test everybody.
Yeah.
And it's crazy because my bad.
It just makes me curious. It just makes me curious. Is this amount of just gumption and energy put into every single subject, or is it only this particular subject where you need hard 100% concrete evidence that this is this? Because my assumption, because I only caught like a little bit of it, is maybe he's an expert in this field. And like that's something that he wanted to go into, which is why he wanted to do just so much due diligence on his own. Because that's the only reason why I could think
he has no scientific expertise whatsoever.
Oh, then nah, like bro, yeah, he was doing the most. He was doing the most in this case.
He's an artist. He's not a scientist.
Here's the thing: it's like you see somebody gets shot with a gun, right? Okay, right? They finna die. They kick the bucket. You go and do the autopsy, and you got someone saying, well, they died because their heart stopped. Okay, let's go into the nuance. Was it the blood loss or was it the gunshot at the end of the day? And everybody's trying to argue that the gunshot, the bullet didn't exist, and the reason why they died is because of blood loss. Everyone's forgetting that there was a event that took place that caused that end result, and people get the context completely lost and mixed up. I don't know how you divide, from point A to point B so many times that you never get to B and say, well, because the math here says that we have to continue to divide, we therefore never reach B, but we're staring at B right now. And we're telling you that's B. And then we keep hearing, okay, but if we got to go to B, we have to go halfway that distance. And then we got to go halfway that. And this is the kind of circular argument. That's why you have to
see the reason why I brought him, I brought AM because AM1, we have had content before, and also that was not related to science. But and we disagreed on different points here and there and stuff, but we just, we figured things out, and it was cool. And so with this, I know he's not a scientist, but I know that he reads a lot of different topics. He looks in a lot of different disciplines. And so when he brought it up, I was waiting for somebody to say, a certain thing. And I was like, oh, he knows what's going on with this because he mentioned it. But now I'm thinking like, okay, I'm giving you the evidence that says like, this is what it is. This answers Koch's postulates, because actually the postulates are just, they're not set in stone. The man knew they weren't in stone when he did it.
Yeah,
when he established them.
Yeah.
And like most of them are like, this is just my guess. This is what I see. This is based on observation. When I went through scientific method earlier, but that study and a lot of other studies right now, it answers all those questions. Like they had controls, they had everything. I mean, for me, that's an excellent study. It did exactly what they set out to do, and it was in a model organism that is easy to get, and it's a model organism that does not even have the gene.
In other words, it's the perfect control.
Exactly.
Yeah,
that's how it was like, and understanding. That's why I was saying earlier in the chat, like, giving time and money to young children for them to, in science, for in middle school and high school, for them to really get hands-on with science. That's what Rodney made a good point about that. They have to be hands-on, and the abstract thinking component of it is I don't know if that's being lost on us or it's not reinforced in school. But that's what I started thinking about, because you literally have to see stuff in your mind happening and then go and look for it happening in real life.
Yeah.
What I mean?
The first thing in science is actually isolating and controlling your variables.
Exactly. You have to do that. If you don't do that, then you can't go anywhere.
Yeah.
And the people that did the study were not like grad students. They were not, they made sure that they weren't grad students. They weren't, because I looked through everybody's names. They weren't grad students, undergrads, high school students, summer students, nothing like that. These were PhDs, MD, PhDs that did the study.
Wow, people with master's degrees. Okay.
No, PhDs, not masters. PhDs.
PhDs, MDs, and MD, PhDs.
My bad. I forgot MD's doctor. Okay, my bad.
I know you're asking questions, and I'm asking questions too, and I'm trying to see, because if I can help somebody along who is not in my field, then then that person has an understanding. Then that means I have an understanding. If I can explain it to somebody who's not even in my field, then I think I'm on my way to doing a good job, with conveying this message, because there's a lot of research going on, like right now as we speaking, that, if I don't feel like I can explain that to everybody, then it's like, okay, do I need to put it out there or not? So I'm not trying to, I wasn't trying to come down on you. There's stuff I don't know. I'm still learning.
I think the tone just came off a bit condescending when the retorts are, well, how do I not? How can I believe this when the government is corrupted? Like little digs like that makes it from a learning moment to a, well, I just don't believe like anything that you're saying is credible type thing. So that's the nuance in it. Like nobody's discrediting you for learning. Yeah, we're all trying to learn, but the manner and how you conducted the interaction, from what I saw, there's always something extra that you added on when you know, either BGS or Nicole would say like, hey, this is the information that I'm trying to provide. It's like, well, yeah, you're trying to provide it, but I just don't know if it's verifiable. And it's like that goes into a whole different like sphere, you know, as far as just like talking points. But yeah.
You know that we just finished kind of dealing with that low key with the flat earth discussion.
Man,
and, you know, I got a lot of similar vibes to that, which is why I brought up the whole, you know, fuel industry. And then I brought up the gravitational waves earlier, because this is literally how that all played out. And you have people on literally the astrophysics level just breaking these things down. And it's a system of approximations. I mean, at the end of the day, and real talk, I don't know why you couldn't take into consideration some aspects of what terrain theory is talking about. It could be an additional nuance. Things don't have to be this extreme one or the other. It's just that in the context of what we have right now, this model fits, it works, and it's predictable, it gets results, and it fits the scientific theory. And that's why you need to respect it for what it is in that context. Otherwise, it's like the illustration I gave before, where you take a snapshot of a cup on a table and someone makes the argument that the table goes on for infinity. And so now, you know, based on the picture, you've got to use all this anecdotal evidence to try to show and prove that, you know, the table does not extend out to forever. But because you can't produce anything that people want to believe, that's the belief that sticks for a while, until someone shows up with a panoramic view camera and they take a picture of the whole table. And then now you get the counter arguments of, I don't believe the picture. But that's exactly what's going on now with the way people can see these things at this point. But people want to stick into that abstract theoretical level and not accept what's been updated in the paradigm that's now shifted because of that.
Yeah. Because I always get, I don't trust my government. I said, okay, you don't trust your government. How about Mexico? Well, I don't trust them either. How about Brazil? I don't trust them either.
I just put that, right.
UK, Spain, Italy, all 200 governments actually believe this. But the thing is, but you actually make it particular to your government that wants to make money. Come on.
Right. And the thing is, BGS gave you a model in order to address the part that you're talking about as far as having a healthy skepticism as far as your government. You look at all governments and then you basically aggregate that information in order to make an informed decision, saying, hey, country A, country B, country C are all saying this. I know that this particular country has this interest invested, this country has this invested, but they're all saying this. So from, you know, you can hypothesize from there and basically cut out the fat or the skepticism that you're trying to acknowledge. But to say like everything is just skeptical and nothing even matters, and it becomes a moot point, and it's almost it's a foolish level of thinking, because it's like you can't do anything with that, because everything is going to have a type of flaw to it. That's the biggest problem.
But you believe in pandemic without evidence. Yeah.
Right.
Right. You see, they don't understand science, and they don't understand that any of us who work at whatever level of science that we work with, we're skeptics. And everything that we do is based off of proof anyway. I mean, we have to. That's why you have controls. That's why you have placebos. That's why you do things in order to rule out error. And see, these people, if you come into it with a healthy degree of skepticism and scientific method where you prove your course step by step, you know, if something comes up that you cannot prove, that doesn't take you in the path that you should go, you stop and you start over. They don't understand that, so they think that the method itself is flawed. And then you have people who basically didn't pay attention. This is junior high stuff, y'all.
Right.
You know, and if you don't believe in science, period, I will never be able to convince you. But then, I mean, look, the whole issue of people sitting up thinking that the earth is flat in 2020, 2019, 2018 should show you that there is a healthy degree of crazy out in the populace. They will never be able to be dissuaded from where they are. Hey.
It's almost hysteria at this point. It's not even skepticism. I couldn't even call it skepticism. It's hysteria, where you're just like, yeah, nothing matters, and everybody has an agenda, so I can't believe anybody but what I feel. And it's like, that's not measurable. Like, you can't really move, you know, strategically with that and make an informed decision based off of that, you know?
If it was ethical, I could prove it very easily. I take him by the neck, and I would rub his face all into a patient, and I would let him see. See, but that's the ex-military in me, because you can only be so dumb, but that's unethical. So I can't do that. And and if you stand them up there and you take them outside of the island in New York and you let them see them burying people in trenches, you take them to Brazil and you let them see hundreds of people being buried a day.
Damn.
You cannot. There's no way for you to refute that. And I'll tell you another thing. A close friend of mine, childhood friend of mine, his wife went on a cruise, came back home, and died of pneumonia. That was in 2019. Okay.
Wow.
All right. So when was the last time you seen that many people coming off of cruise ships? And you know how black folks like to cruise now. When have you seen that many black folks coming home and dying of pneumonia? You know how many people die of pneumonia? I mean, look it up. BGS, if you get a chance before all of this took place, check and see how many people died a year of pneumonia.
Okay.
Rodney, they didn't die of pneumonia. They died.
Absolutely. I don't have the numbers, but you know what I mean.
It's the 5G.
Deep brain didn't have enough oxygen, so they died.
Well, I mean, if you look at the screen, like this is just an example of a gel of the thousands of gels I've run. So when they were talking earlier this year about how they were using a RNA kit to isolate RNA, or not even isolate RNA, purify RNA from the virus and then do what they had to do. Okay, sorry about that. And so what we do is we isolate RNA from the cells that we grow. We reverse transcribe the RNA into cDNA and we run the cDNA on a gel. This is a 2% agarose gel. They did the same gels when they were analyzing patient data. So if this is patient number 1, patient 2, and patient 3, you see differences in the way the bands look. So you can say, like, if this is a ladder, this is a control, the standard ladder. The brightest band here is 500 base pairs. The brightest band here, 500 base pairs. I just know that. That's 100. That's 100 right there.
Yeah,
so anything like 100 base pairs. So this is like 200, 300, 400, like, so these 3 patients, if this was 3 patients, they would be 3 different patients with COVID. Let's say COVID is going to be right here. And then you got COVID. This person has COVID, but less. This person has COVID here, but not really in these samples. Maybe these are different samples, like heart, brain, lungs, spleen, kidney. You see what I'm saying?
The genetics do not lie.
This is not COVID, but I'm just saying this is from prostate cancer patients, but still,
right. And there's no way for you to fake that, and that's what they don't understand.
You can't make that up. That's what it is.
You can't make RNA into something else. You just can't.
No.
I mean,
it is what it is.
I mean, it's so frustrating. I'm telling you,
it's so many steps that go into getting to that point. That's not even the end of the experiment. That's the middle of the experiment. The next step is to take those samples, cut the gel out, isolate that, and purify the gel, purify the DNA out of the gel. That's another step you can take. The gel is like literally like a hard piece of Jell-O, and you can cut those little yellow marks. You have to wear a shield because it's UV. That's the only reason why you can see the bands is because it's the UV dye in the samples when you load the samples on the gel. So you can see it with UV light. Well, I can't put my hand over the UV light, so I have to wear the gloves. I have to wear the PPE and everything just to cut the little strip, the little gel band, to cut it out, isolate that piece of DNA out of that gel, run it on another gel to make sure it is DNA for real. And then I take that and quantitate it with real-time PCR. That's what they were doing with the patient sample.
Yeah, multiple confirmation. Multi-confirmation.
You confirm expression 3 times when you do that. 3 different times.
And with 3 different methods.
3 different methods, 3 different samples, 3 different patients. Each patient is you have to take sample from each patient at least 3 times.
Yeah. In fact, the average samples that I was reading, the average samples that CDC gets from from flu is like 77,000 per year. I said, you got 5 million samples that going to the CDC in 6 months on coronavirus. I mean, 50 million, samples, positive, negatives, 4 million that are positive that have been verified, because they have to verify this. It's not something, it's not an estimate. They have to verify each case because people are so pissy about this. They don't do that.
They don't do
get
that for
this.
everything. They don't. They do like sorry, Ronnie. One.
Oh, you're fine.
Thing. Last year, I know for a fact they didn't do it for every case. They did it for like every 100 cases. So the fact that no one had no questions about that last year, and then they did it differently it wasn't even consistent from state to state or county to county. It's like one state will do it, or one county will do it like every 100 cases. Another county do it like every 10 cases. Like it was just ridiculous. They had no consistency. So therefore, when you compare the flu last year to COVID, there's no standard to compare COVID to, because they weren't doing things right last year.
No.
Right.
No.
Right. And they're going back. Have you guys heard about? They're going back and they're taking tissue samples from a lot of the unexplained deaths last year, like the pneumonia, and they're testing those. So there's a backlog on even the dead people that they couldn't even explain from last year.
Exactly. Yeah, late fall. Yeah.
Right. So once this all clears, we may find out that COVID was here in maybe mid 2019.
That's what they're saying. That was late, August or September. That's what they're saying.
That's what I thought.
Here's the logic that's being presented to counter that, which is, how do I trust the process that information was gathered under? That's all I derive from that, and it's frustrating because even BGS can contend to this when he was speaking about Spanish flu. What was the condition of the people who were literally carriers and dying from it a lot of times? Did they show symptoms? Were they not?
Yeah.
So like, you can't use your subjective experience as an indicator or reflect it and project it out to the entire situation. Not when you've got something that is an RNA strand, it's subject to mutation epigenetically and just naturally. It's literally how these things are tracked.
Yeah, and everything they said that this virus would do just about is it's doing. But the thing is, it's brand new. They've never seen it before, so even the scientists are guessing. They're learning as they go along.
And I think that the question is for the detractors. This isn't just like poking on AM1, because I don't think that he's considering himself a detractor. If you've lost confidence in the system and you're basically the methods in which they're applying to try and solve this problem, the question you need to ask is, what method do you use to trust it?
That's what I was going to ask.
What's it called? I provided an analogy, like, so if Donald Trump says that gravity no longer exists and that is fake news, does it make it no longer true and unverifiable? Like that, like certain things, like if you don't trust, whatever system or, whatever method that is in place, what is your alternative? Are you going to test it yourself? Are you going to, with the example you gave, Nicole, as far as the UV, right? If you believe the UV isn't going to burn your skin, right? So are you going to use your hand? You're going to use a stick, or is a stick no longer a good measure because a stick isn't an actual human hand and it can't replicate skin being burned? That's my question. That's my question to the detractors. If the model or system that's in place is failing you and you think that it's no longer verifiable, what is your method? So then we can, start talking. Because right now you're being an armchair critic with no engagement, and that's the part that's frustrating me. Is when you have folks who are experts in this and like this is their life, this life works and studies, and they're giving you evidence, but you're just sitting there just saying, well, I just feel like this is no longer true because of XYZ. And it's like, well, let's hear your input. Well, I don't have anything.
Yeah, they're gambling with no money is what they're doing.
Right. So then now, go ahead.
Go to the same place. This is affecting people in a way that, you can get folks killed. Absolutely. And that's the difference between this and some flat earth stuff. And that's why this needs to be taken more seriously. Right. Because it's how this misinformation spreads when I'm not directing this at AM1. I'm gonna direct it again towards people that want to blindly disagree with this off principle of, you know, their personal beliefs. You need to recognize that if you are rejecting the way in which the information is being presented to you, and you're not taking the time to educate yourself on how the process even works to be qualified to disqualify it, you're really just literally misinforming yourselves and other people. You can't go off on your feelings with this.
But it's not just them. I mean, look at this stuff that just went out. Brett Barb, walks out a bunch of witch doctors and basically have them, run for 24 hours, and then the next day, Twitter and every other reputable, source removes it and tells you shouldn't believe it. The damage is already done. And I'm gonna tell you, the AMA, they need to go out and they need to start recommending that these people lose their licenses. If you're a nurse, if you're a doctor, if you're a scientist and you go out with some quackery, you need to lose your credentials.
Yeah, I agree. I got the email from you, West Coast African, and what Rodney's talking about with what was her name? The lady with
the Nigerian doctor.
Emmanuel?
Emmanuel. Yeah.
She's talking about exercise and succubuses. And
I saw
dead
that.
Yeah.
I had to send it to you.
She has a church called Fire Power Ministries.
Yeah.
I was looking at a little bit of that, and I was like, wait, what? Like, I don't know.
Yeah, I had to send that to you. Sheila Emanuel. Yeah, she's talking about mixing hydrochloroquine, zinc, and Zithromax all in 3. It's like she treated over 300 COVID patients, and she was saying all types of claims, saying that she said nobody in our office was able to wear masks, stuff like that.
Now get this, Nicole. Now I'm telling the truth. Now get this. If she is a doctor, then she should know that azithromycin is not going to work on that. It's
that's something they get standard. It's a Z-Pack. They just give that standard food and flu and stuff.
Antibiotics don't work on viruses.
No, that doesn't. Yeah, I was like, wait, that's antibiotics. So
antibiotics don't work on viruses.
That won't work.
And hydrochloroquine is pretty much it's parasitic to the body. Basically, what it's going to do. I mean, it used to be used as a chemo.
Okay.
An alternative to chemotherapy. So basically, what you're doing is you're going to be destroying rapidly producing cells and causing probably some of the same symptoms that you would have in COVID, like the loss of taste. The lining of the mouth starts to slough off. You're going to start causing problems also. Not to mention, I mean, those are used for autoimmune diseases. So the treatment doesn't even go with that. You know what I mean?
As far as mostly for like parasites and stuff, hydroxychloroquine is something that you're supposed to take something with it as well. I forget the name of it, but it's supposed to be something that's taken in addition to as a supplement. And you're supposed to change your diet. You're supposed to do a lot of things. Get rest. Stay well hydrated. You're gonna
have to take something for the inflammatory process that takes place. So you're probably gonna be on steroids.
They have a non-steroidal, or excuse me, not non-steroidal. They have the steroid. What is it? Glucocorticoids that they're studying right now. And some of the steroid treatments, I know there was one they were saying that it's the potential snake oil. They have this steroid treatment. It was on Fortune and a few other websites I was looking at. But they were saying that this miracle solution is probably just snake oil from a Texas doctor. So I think she isn't the lady, Dr. Emmanuel, isn't she in Texas as well?
I believe so. I believe so.
It's a lot of mess going on in Texas. I don't know what to do.
Man, don't ask me. They crazy out here.
I don't. Texas is Texas. I'm telling you, Texas is something else. Something else.
I don't know how to spell adept because see what they say zinc. I mean, I take everything anyway, but then they say zinc. They don't give you a like Rodney. You made a good point one time. You were saying, you can take take all this extra stuff, but you're gonna have to flush it out.
Right.
Your body only needs a certain amount, and it's going to need more, of course, during an infection, but it's not going to need like 8 times more zinc, or anything like that. And my thing is, if you want to really get the vitamins and minerals that you really need is from the food, because one of the symptoms of the virus is it's going to reduce your appetite. So making sure that you're eating properly, getting the nutrients directly and not from a supplement so much is very important. You know,
and hydration. And if you're taking in,
staying hydrated, because people are saying that constipation is one thing, and they're not drinking enough water and getting enough electrolytes. That's another problem. You're not flushing your system out consistently, you know?
Shouldn't this be where terrain theory comes in that regards, you know, to explain how something bad being introduced can be aggregated that way? Not necessarily that's the cause of it, but, you know, this is how people can approach certain levels of preventative things. You know what I'm saying? Because everything you mentioned sounds to me like you're providing your body with the means to address these things on a natural level. And that, you know, it could be that a lot of people already in negatives with a lot of these things, not having enough vitamin C in their reserves anyway, because they're dealing with other issues. They already have inflammatory situations going on because of foods they're eating, and a jacked up biodome and their stomach, the gut health and all of that. And you know,
we have ways of checking all of that. In the beginning, you get blood work. You work a person up, you do a complete workup on them. You check out and see how they are metabolically. And then, you know, like you have to know that if you super saturate the body with vitamin C, guess what? It's going to piss out. You're going to piss out about 80% of that, which that has to be filtered out through your kidneys, which you are overworking your kidneys, which the COVID is going to also overwork your kidneys. Also, you're already at risk for renal failure. So if you're already at risk for renal failure, why are you going to increase that risk, you know, 100-fold by adding a whole lot of extra stuff that your body's not going to, look, metabolism is important. Okay.
Right. That's what I meant about eating properly, not necessarily supplementing.
But it's more than that. You know what I mean? Even if, some things have to be concentrated because you can't eat 500 bananas. You know what I mean?
Well, that's true. So, I just mean that, you know, compared to the fast food diet, a lot of folks got. You know
no. We don't. None of us are advocating that at all.
That's all I'm saying. Because we don't know who's listening. I'm just saying that.
Everyone can't afford the food either, and so getting the supplements is important for some people too.
And that's why we're dying at the rate that we're dying.
Yeah,
I agree with that.
It just kills me when people all of a sudden they want to get, you know, like they want to start taking vitamins and supplements. I'm like, what was you doing the past 20 years? No. You was going to KFC and you was going to El Pollo Loco and Del Taco. Okay. Now I do love some Del Taco. I ain't gonna lie, but I will say I don't go every day. You got people that's like going getting their tacos. I gotta get my tacos every day though. Everyday food? No, you don't. Like they would go that's what they. Then all of a sudden the COVID hit, and I was like, man, I gotta get my supplements up, man. I don't feel so good. Really though? Come on.
Nicole,
now that's the perfect experiment right there that you see is not going to work. I always take it, I'm not saying don't take any supplements. That's not the case. I'm saying like don't depend on them, because that is a major symptom when you have a virus that's preventing you from wanting to eat, from the desire of eating. It's affecting your brain. Okay, it's affecting your pituitary gland, perhaps. It's affecting certain parts of the brain that should trigger you to want to eat something. The satiety, mechanisms that are in your brain, neurotransmitters that are telling you, hey, you hungry, go get something to eat. The GI tract is being affected. If, salivary glands are being affected. If you see food and you're not hungry, you can't smell it. You probably can't taste it that well or anything. So that's not good. So making sure people that I was telling my friend the other day, making sure that she goes to her mom's house, just check on her. She's positive, but she does not have symptoms. I was like, you need to keep checking on her. She's an older person, and older people don't like to eat anyway. They, eat like little birds. So, yeah. Hostile that said lift weights. Yeah, if you can.
I'm vegan, so I know all about that. I have to look for different alternatives of certain things. So I have a lot of experience with that. But, and I'm not saying that to advocate anyone being vegan, because that's an individual thing. The point is people need to improve the quality of the foods they're eating and pay attention with regards to that. It's an over time thing. You know what I mean? Just people just did that much if they could, then it'll go a long way. You got a lot of folks that don't even understand, how carbs, proteins, fats, and all of that, and how your micronutrients all play a big role into every system of your body. And just basic dietary stuff. I mean, even folks in the medical field don't really get a lot of opportunities to talk about that. And, there's whole arguments going on right now about, the food table. And, and that's leading into some of the things that AM1 was talking about with regards to, profit-driven sales, with regards to the studies that get funded that greenlight a lot of government policies with food and stuff like that. But, hopefully this is a wake-up call for some people. This needs to be taken more serious than what it is. And it's more inclusive with regards to lifestyle and the food you're eating, and your basic safety practices like wearing a mask and just the simple stuff like that.
I was just looking at my Facebook and somebody's got wearing a mask is ritual, but it's not science.
Just go.
Shit is what I'm talking about, man. Like,
oh, Jesus. Lord. Help us.
Yeah, I wanted to hear, Salchi, what you had to say. You on here quiet? And Albert, I was
trying to sneak in. No, I mean, the first thing I want to say mainly is I happened to speak to my cousin on Sunday, and we're both really healthy, go to the gym, practically almost gym rats. And of course, he didn't take it as serious. I mean, we live in different states now. But he told me he contracted it from one of his domino partners, and I think 4 or 5 of them normally play. And apparently, one guy went out, I guess, the night before, and of course, they went and played dominoes, and they all got it. But of course, he's healthy. He's healthy, but he said the only thing of course, he saw his doctor, but the only thing is that he had like the chills or something like that for one night, and that's it. And then now, like when I was speaking to him, it sounded like he was like a cold was clearing up, and that's it. I guess because he's healthy, that's it. But I mean, the way he was talking, he was like, "Sal, it's not that serious."
Did he receive any kind of treatment?
Nope. I asked him, "Did you get any treatment from the doctor?" He said, "No, I'm not going to give him anything."
So he doesn't know what's going on with him. And COVID, y'all gotta stop looking at COVID as like something you're gonna get over. This is gonna be a long-term situation.
Yeah.
Because COVID patients are losing a substantial amount of lung function associated with this. So he doesn't know what his SATs are. He doesn't know. He assumes that he's healthy. But I have another situation where there was another bodybuilder that got it. He lost almost all his musculature trying to fight that off. And to some degree, from years of seeing people go through different types of debility, basically, if the amount of musculature that you have and fat that you have will determine whether you can get over a serious illness. People who are really, really scrawny, sometimes they don't make it. You know what I mean? So, because your body's going to break down some of that extra that it's been storing for a rainy day. That's how it works. And see, a lot of people don't understand that part of anatomy, and physiology, and, if you're really, really lean, that doesn't mean you're going to make it.
Yeah, you can be lean, but you may not be metabolically healthy.
Right, and your body's going to break down your muscles, and when it breaks down that muscle, what's that muscle going to turn into? It's going to be protein in your urine, which is going to put you into renal failure.
Yeah, put you in big danger then.
Right,
kidney issues and all that.
Right.
Yeah, see, I show what you're talking about. A lot of things that you've talked about, Rodney, and I've heard other people discuss. I mean, I literally show like the same organs that you all talk about. They were in the study I just
absolutely. Are you seeing tissue samples from the heart? Yep, absolutely. That they're finding stuff in heart tissue, lung tissue is showing up in liver, in the heart,
the brain,
in the spleen. Absolutely, and that is crazy. It's like it's breaking down this tissue, and the tissue staying intact. It's not being metabolized. It's just floating throughout the system, which is devastating.
Yeah.
Because it can cause the same kind of damage as a clot. It'll just destroy what it lodges into.
That's what you saw on the lung tissue. That's what those images were. I mean, people watch it later, they can rewind back. But when I was showing the figure, it had like all the lung, like the pink, like hot pink, purple looking color. That was tissue from the lungs of the mice. And the white, any kind of white space you see, that's a hole in the lung.
I can't even imagine what the
that's due to. I didn't say what it's due to, but you know, Rodney, what it's due to. If a blood vessel dies, then you have this huge space where there's no tissue, there's no vasculature in that area anymore, and it can't repair because that was after 8 days. Well, they went 8 days because they were dead.
Right.
And 8 days for a human, or if for a mouse is 8 days, for a human that's about 30 to 34 days, depending on the type of mouse. You know how strong they are. I mean, that's for a human, that'd be like a month. You're dead. So, you know, 8 days is a long time for mice after getting that much injected into their nose, and it went to the brain because of the injection site.
Let me tell you, I've had people that died in 4 days.
Wow.
4 days. And then I've had people that have lingered for a lot longer than that, and they did not, it wasn't a good situation. I'm telling you. So all these people that stood up, I wish that these people could see that, because they're not showing that, because they're not showing it because of you know, a lot of different reasons. You know, nobody wants to see a person suffer, you know, and you know, I'm telling you, man, that this shit is, I mean, I'm sorry, I mean, I can't tell you, I have been around a lot of stuff throughout my years. I'm telling you, I should be a lot more terrified than I am, but I tell people, this is what I tell people. I tell people, if you are comfortable when you're working with this, you need to stop. You need to get out.
That's true.
You know, you can never be comfortable, or you're gonna mess up.
Yeah, because last night we had one of my subs on. He said his sister, who had her rebreather stopped functioning for like 3 minutes, and she caught it.
Yeah, because it was circulating bad air.
Circulating bad air. Yep. 3 minutes.
Yeah, I heard part of that stream, part you were talking about when you came on, Rodney, and yeah, that suit, like that's just because you don't know until you know it's done. You know,
ultimate bad situation. But
it must have been in the suit, you know, already.
Yeah, and you're supposed to. And anytime you have a suit like that, you got to have fail safes in place. You know what I mean? They should have been able to. It should have shut itself off and contained the air that was already in it, so it gave you enough time to get outside of the environment. I mean, so I don't know how new this suit is, but I've seen suits like it. So I don't know. It could be just the atmospheric portion of the suit that may be so new that it just wasn't right. But you know, who can say? You know?
Yeah. You can. First thing I thought was the filter, and second thing I thought was tear.
Right. That's what I was thinking. Either it's a tear, but then if it was a tear, and then it you know, it detected the tear, you know, it still probably circulated bad air for a minute before it shut itself down.
Yeah. And shutting down is, you know, shutting you down. So you got to get out. That's why you saying that she was running, trying to get away. But you're running into a contaminated area. Any way you turn is all contaminated. It's not like you can jump down a laundry chute. Every way you turn is contaminated.
It's there.
You can hold it, try to hold it closed, but it's like it only takes a few seconds. And you know, it's really like I was like, oh my God. When I heard that, I was like, this. So like for me, I've already been in that type of situation, wearing that type of suit, not for this, but for another situation. I have to be in a clean room to work with gold nanoparticles. This is totally different. And so like, you know, when I love being in a room working with gold because I like shiny stuff, but at the same time, I was like, you know, I gotta wear this bunny suit to be in here. I mean, it was a lot of gold, you know. I was like, wow. And they were, you know, pulverizing it into nanoparticles, and we can see it under the microscope, you know, and all that. But I wasn't worried at that point. Only thing I was really worried about was the dust, you know. And then when we got out, we had to go through the mist steam, corridor and everything, and then get out. But in that situation, doing people's lives, I was like, oh my God. I was worried about just inhaling some dust, and that we can't even see. These particles are so small. But with her, like, I hope that they keep an eye on her. Definitely.
Right.
I really
That's what
hope
I'm
they keep an
saying.
eye on her, because it can switch overnight.
Yes. And I'm telling you, some people scratchy throat, and by the time the respiratory symptoms present, it's kind of too late. I mean, you can be satin in the 70s, and I'm gonna tell you, anything below 90 is bad. 70s is catastrophic.
Yeah, you can pass out walking down the stairs.
Right, and then after that, you're gonna start having tissue damage.
Yeah, because things will get to I think that's why those images look that way now that you said that. But the inflammation those mice they had tissue damage, loss of oxygen, that was all affected. And so if they can show that, and then I wanted to see
hypoxia, and then you'll start having tissue necrosis.
Exactly, necrosis. And if you have a lot of necrotic tissue that is hypoxic, it's not getting enough oxygen. It is lower than 2% oxygen in those areas. Some cells will try to resist that low oxygen level, but other cells can't, and they just die out. So with that, you have a hardening of certain tissue. You don't want hardening of your lungs. No. You don't want hardening or plaques forming in the heart. Hardening of tissue is not good. When it can't move and contract, you don't want that, because then now you have a heart attack. Massive coronary or something like that.
And we don't.
Now you got a stroke.
You don't realize how much elasticity is important to to certain tissues. Especially heart tissue, especially lung tissue. Your entire thoracic cavity has to have some elasticity to it, in order for it to work properly.
Yep. And all those things, like, only thing I think that paper needed was, and I think they mentioned it in the limitations of the paper, was they didn't have they couldn't get access to patient tissues to stain them for the ACE2 with the ACE2 antibody and compare the tissues to what they saw with the mice. That was the main limitation I think they had, because that would have been, well, I don't know if that would have been compelling. It would have been compelling to me, but I don't know if other people who, may be skeptical and questioning, if they would see that as compelling. Like, that would have been like, hey, that's it. You can recreate this, and you show that it was very similar in the humans and the mice. Shit.
Yeah, but you've got an experience. You've got experience seeing that correlation.
Yeah.
So
it's a 1 plus, like BGS said earlier. I almost called you Dr. BGS. It's like you said earlier, BGS. It's like it's 1 plus 1 is 2. Like, that's it.
Yeah. Right. But that's why.
But if
that's why
peer
groups are important. That's why you don't sit up and have a bunch of doctors go and try to prove their findings to a bunch of retards. You don't do that.
Oh, yeah.
But what I'm saying is, people have different definitions of what 1 and 2 even are. And when you're that far away from a point of trying to have a discourse, this is what you run into, is all I'm trying to tell you. That's what I'm saying. That's why you have to have some kind of pre-101, just some kind of basis. And like you mentioned earlier, Rodney, and actually Nicole said this earlier too, schools are not being provided enough to really present this to people in a way where it sticks. And so when you get into these topics, and people are left to their subjective opinion, and then when your subjective opinion has a big influence on a consensus-based dynamic that they get on social media, what do you think happens? You get a particular viewpoint that gets literally hyper-projected into a greater scope of people, and this is why this thought process continues to show up, and y'all are pulling your hair out, literally trying to present basic facts to people.
Because it's philosophy. It's mutated into a philosophy of reality. Well, what is actually real? What is actually tangible? And I mean, you cannot do that. I mean, look, no, you can't do that. Some things are universal. Some things are there. That's why we have laws in physics, laws in math, laws in science.
Yeah. Well, the thing is, what I don't understand, Rodney, is that, okay, say you don't believe the press, you don't believe the doctors, you don't believe the stats, but the thing is, you have an expert that that you supposedly trust, Okay, like Nicole. Nicole is showing you through her expertise, and you can trust her because you know her personally, right?
Right.
You can see, like, I'm not gonna lie to you, because I can tell, and you know why I can say that with confidence, because I just showed, like, what was the video I just did, like, where they try to pass off some bullshit in a publication because of nepotism, and they want their paper published. I can show you, like, if it was some bullshit, I would have came on here and said this some bullshit. I would have told y'all, hey, look at this mess they tried to publish. They try to get out here. I would have said that. Like, I can see the BS in the papers when they have them published, and a lot of times they have to get retracted, and they lose. You know, they can't publish for a year or something like that.
Good.
You know, they lose their
should,
they lose their
and they
labs.
should
Livelihood.
post that up and make sure that people know that they tried to pass off some bullshit.
Yeah. If it was some BS, I would have been like, okay, I'll look through everybody and see. Like, but my thing is, some people will look at that article and they would say, oh, well, it said University of Washington. You know who just funded University of Washington last year? Gave him $2 billion? Bill Gates. What the hell they got to do?
Yes. Who cares where the money came from?
Because that's where he's from. Of course he will give.
Hey Rodney. Hey Rodney, man. All universal laws are alleged because you know everybody's corrupt. Everybody has an agenda.
I know.
Gravity.
Everybody's corrupt.
Trust no one.
Yeah, I know. I'm the most corrupt person up here. So
right. Some of the arguments, it's like, well, you know, I mean, is gravity really real? Yeah. It's like, well,
you're looking in the comments. Yeah.
Is gravity really real?
I'm really not. But I mean, I just try to stay focused, man. I mean, and it really, you know, I quit on myself. You know, I was actually headed to medical school, man. I had my son, and I focused on raising him, and I stuck with nursing. And you know, I should have kept going. You know, but I didn't. But I still know what I know. So, you know, people who don't like science won't have the affinity for it to be able to make connect the dots. You know what I mean? You can connect the different things. If you can't follow a disease process, you will be no good in medicine.
And basically following the disease process is just logic. It's not
right.
It's not science. I mean, it's not rocket science. It's basically following the evidence and where it leads until it stops.
And that's what I tell them. It's like mechanics. If you're a mechanic, it's just like, imagine that car is a person. You know what I mean? It's the same thing. A lot of the things work the same. You know, it's not that hard to logically make, connect dots.
But whose logic though? Whose logic though? BGS? Because everybody's corrupt.
Everybody's corrupt, but I don't trust the white man. But he's the one that's defending the book.
Right. Scientific as per their definition, which is why we get into this whole I feel and science using the same sentence, and it makes me want to pull my hair out because now you're being philosophical, just like Rodney said. Can't nobody tell you what your philosophy should be. And then here we are again with the whole subjective opinion being cloaked in scholarship. And it's like you have to concede at some point basic truths and use that as a basis of communication. That is how communication works. Period.
Well,
I think it's just being intellectually lazy. That's right. And I mean, I did take some of these courses in college, but I could just go back to high school taking chemistry and biology. I think that's enough for you to get a clue.
Absolutely.
I think
that's all you need, actually. Enough. I mean, if you don't understand that, and if you haven't had that, then it's hard to build upon that. It's just, those are the basic building blocks. Like what you just said, like Salchi, I use that every single day. Like basic understanding of high school biology, chemistry. I can't stand chemistry, but just the basics of it is what I use every day. And I go in there and I always say to myself, like, I'm doing this experiment. Would I be able to explain this to a 7-year-old or a 12-year-old? Would I be able to explain this to my nephew, who's 11? Like, how can I explain this to them? Like, would I just present it? Would I show the gel earlier? That's like, that one gel is like, two parts of a figure, but it needs to be explained to a kid for it to make sense to anybody.
But you know how they explain that same gel, diagram that you showed is basically basic genetics. You have to perform an experiment.
Yeah.
With basic genetics that does that.
Yeah.
You know, so that's not. It's not. I mean, if you paid attention, you know what I mean. I still remember that.
Yeah, that gel was I made the gel by hand. Like I poured the agarose powder, I poured the TB buffer, I warmed it in the microwave, I poured the gel, I put the combs by hand, those little holes that you saw, those were where the combs were. So I loaded the sample with the pipette tip in every single well. Every lane is I had to load from a different tube. So I have different samples and they give different levels of expression of that gene. That's why they look, some are high, some are low, whatever. But I didn't know that before I ran the gel. I didn't know that sample number 5 and sample number 12 were going to look the same.
Right.
You know, I was like, well, sample number 5 is this treatment and sample number 12 is that treatment. Let's see what happens. And I was like, oh, okay, it's the same. Well, that drug didn't work. Moving on. Like, you know, that was my conclusion. The drug didn't work. I'm moving on to drug 1 didn't work. I'm moving on to drug number 2. Like that was it. It's not hard. You know, it's very elementary, but you have to like big concepts. I notice people who have these big concepts and it's hard to break them down.
They left that?
Then I start questioning.
Last night I pulled it and put it in there.
Take a picture.
You know what I'm saying?
Take a picture.
Don't leave the stuff here.
I agree. Yeah, take a picture. And matter of fact, they don't need to leave here until before they talk to you.
Yeah, I didn't know I could be back in the still now. You know, you had this stuff.
Right in with a handling business now.
Right.
I'm gonna cut this off, y'all. I know y'all got things you need to do, such as dream about, you know, the corruption and
Thank you so much. Thank you so much for having me on.
So corrupt.
I really appreciate it.
AM1, you hear this? You better not come at me. AM1, you better not.
Was he gonna say? Was he gonna refer back to the video?
No, we can have another stream about it because I know we want to get some other things. You know, I gotta give a person a chance to read through and see what's going on and but now we know like where we stand on different things and that's fine.
Yeah, more science panels.
Right. I was going to say, we need more of these so that
especially during this time we need more science panels.
Yeah, definitely.
So people can have a point of reference because, like you said, you're a subject matter expert, and so here you are with the 2-for-1. You know, we can take the information you present, and there's studies and there's ways people can elaborate on research so that we can have a basis to counter a lot of these real simple misinformation dynamics that people throw at us on the top, and it's just. It's needed, and I don't know too many places to go. I mean, if I didn't have my mom who was already in the field dealing with this personally, there's a lot I wouldn't know. Because if I leave social media, what am I going to be left with? You know, for all I know, reptilians did this crap. So it's like, you know what I'm saying?
It's not the iguana.
Right.
Yeah.
Nicole, listen, they too woke for us out here, man. We need to come in and say,
you gonna stop, man. You know what? I'm gonna have to find some more information. I have a list of different topics to go through. A lot of them are not about COVID, but you know, it's like related to other things. But I think having a stream, I'm trying to do it like maybe once a week or whenever people want to, like have one stream and then a follow-up later on that week, because I feel like people need time to digest the information.
Right.
You know? And like AM1, you can read, you can, you know, don't let what people are saying like on the media or whatever, don't let that like limit what you want to look at. I look at all kinds of crazy stuff and I'm like, they're doing research on what? Like I'm reading and because I just want to know like what are they doing out there. But I appreciate y'all coming. Thank you, Shango, for coming.
Anytime. You want to get in touch with me?
We won't miss this showdown.
Yeah. Yeah, if you want to get in touch with me for the stream or whatever, man, just hit up BGS. He knows how to get me.
Oh yeah.
Okay. Yeah.
You're actually very kind on Nicole. You're kinder than I would have been.
I'm not gonna go off on them, man. I'm trying to give somebody, you know, a chance because I remember when I was starting, I didn't know I didn't know. Well, I was like,
Yeah, and I understand
16,
him wanting to
17.
question things, but sometimes you got to go to experts.
Right.
Yeah,
Right.
you have to, and I don't know. I'm still learning too. I'm trying to see where somebody is, what they have experienced, what they've read, and then either it matches either it matches and it's logical or it doesn't. Like that's how I see it. And I'm not gonna yell at him like he do his thing and I do my thing.
Finish him.
No, she did a good job.
I'm not gonna let that part of me come out.
I wasn't gonna do bro like that.
Exactly, Gino. He said BGS too kind too. Exactly. And then you was clapping the other day. I was like, what? He clapping in the background? What's going on here? I'm trying to get you to understand. Ask the question, goddammit. I'm like, what?
Yeah.
I had to rewind that part. I was like,
yeah. No, they were butter dancing, so.
Butter hot, man.
Bosco
step, man.
That's the new name for Coon. We call him Bosco.
Yeah, Bosco.
I heard that. I was like,
oh yeah.
Oh, you mean like that dude this morning? I got a little bit of it.
Yeah. Put down Bosco. Put down the biscuit, sir.
Yeah.
Right.
There's a better way.
Facts.
You know it's gonna be trouble when BGS say, "I'm gonna ask you one more time."
Right. No,
you should just go ahead and just be like really, really listen, like like really listen.
Break everybody down. That's when you know somebody in trouble.
Oh yeah. That's when you're like, let's everybody mute up.
I am speaking English, aren't I? Let me ask you one more time.
Yeah, I don't want to go off because like I know how it was and I was learning, and I mean one day I might go off. I don't know because I'm trying to get better, like not mute myself and be like, whoa, like just mute up and go off. But I might go off one day. It depends. If it was one of the people, I would have went off.
You're actually really kind.
Yes.
Yeah, I just I appreciate that. I just don't.
And really patient and really showing the slides, and we're gonna go back over this one more time. See this?
Right.
See that? See this?
But mama's a teacher, so
how we know that's paper we looking at, cuz.
Yeah. Oh God.
What about the terrain method? Okay, how do you transmit? How do you transmit the process?
Yeah.
In other words, you couldn't answer. Well, I don't really know, but so in other words, you're a self-contained entity. How do your exosomes get out of you and into somebody else?
One hop this time.
Yeah. Do they? Does he understand vectors?
No, I don't think. I think they went over his head when I said vector.
Oh,
there's vectors. There's the vector thing. I mentioned that a few times too because that's what they use, the adenovirus vector.
He didn't get it.
Yeah, wow. Vector is just what you need to get from one place to another. It's like transmission.
Yeah, they would assume that you're transmitting something. Okay, so how do you get similar circumstances with two different people?
Right.
Well, if they're different continents.
If the same environment, what if they're not in the same environment? Well, we have to find out how come they're exhibiting the same symptoms. Okay.
They got the same mold. Same dirt on the ground, good. What you mean?
It'll be okay.
And that's why you contact Trace.
If I can get one person that that understands that can kind of like come to the I don't say to come to the light, but come to the darkness.
We're gonna start calling Carol Ann. Come to the light, Carol Ann.
No, don't go to the light. Come to the darkness.
Y'all won't mess around and have people
then that'll be good.
Listen, man, I think that the Bosco step is turned into the DJ Casper slide based off of two ups, slide to the left. Now, how many jobs are available? Enough. How low can you go? Can you go down low?
Right. But I'm gonna do another stream this week. I have to do one on Thursday. Probably around the same time.
All right.
So and then I'm thinking maybe one this weekend as well. But it was good having y'all here. I appreciate y'all.
Us too. Thank you so much.
All right.
Good night too.
Yeah, appreciate you.
Y'all have a good night. Peace.
All right.
You too. Have a good night.